हाँ।
पता है तुलसी में कितने कुदरती गुण हैं। ये गुण चाहिए तो पानी पियो। और लौंग के गुणों के लिए जीरा, पानी और पत्ती, पानी, यानी अच्छी सेहत के लिए। মাঝিকে ওই ঘরে, একদম ওই ঘরে নিয়ে যেতে বলো। মাঝি ওই ঘরে চলে যাবে। দাঁড়াও দাঁড়াও দাঁড়াও, পায়খানার মত এইখানে হাত দো। এই যে সাবান আছে, এই যে সাবান, এই যে সাবান, ভালো করে হাত দো। আর হাত ধোয়ার পর ভালো করে ধোয়ার পর তারপর আমার কাছে এসো। বাবা তোমার এক একটা কথা কিন্তু এখানে চলে যাচ্ছে।
বলবে, তুমি এখন কি কাজকর্ম করো, তোমার কোনো অসুবিধা হবে না।
Good morning, sir.
Hello, good morning.
Good morning, sir. Good morning, sir.
Am I audible?
Yes, sir, you're audible, sir.
Okay, thank you.
Sir, it's 9 already. Should I start now?
Yeah, please, Tariq, you can start. Please.
Okay. Thank you. So, hello and good morning. Welcome to the two-day international seminar on the world of plants, its impacts on indigenous people, society, and biosphere, organized by the Department of Botany, Government General Degree College, Liverpool. As we know that in this time of crisis, in this time when the global spread of the pathogen has brought the world to a halt, the academia has actually been led to look beyond the conventional chalk and talk teaching model, and the technology-based practices, online learning, digital communication have become the new normal in academia today. However, if we look the other side of the coin, with the outbreak of the pandemic, all sorts of borders, the borders of bodies, the borders of home, the border of nation, space, and the race itself are broken open and subtly punctured. This breaking of the boundaries is important in the sense that it deconstructs the myth of hierarchy and privilege. That a young college like Mohanpur Government College, which is located in the far off border area of West Bengal, is organizing this kind of academic program with globally acclaimed academicians as the resource person is a positive reflection of the breaking of boundaries that the virus invasion results in. We are not sure whether without this boundary breaking and normalization of e-conferences across the world, we could have done it or not. Charles Dickens has rightly said once, it was the best of times and it was the worst of times. So we are organizing this seminar because of this boundary breaking. I, on behalf of the seminar committee, the organizing committee, welcome all of you on this virtual platform. I am Sheikh Tariq Ali. I teach in the Department of English in Government General Degree College, Mohanpur. So before we start our session today, some provisions, as this is a virtual platform, we must keep in mind some technical thing. Number one, please, I would request all the participants, do not unmute yourself unless you are one of the speakers. All queries can be posted in the chat session. You can turn on your video if you like, and you can unmute your microphone when you will speak something. So with these words, I now request the organizing committee secretary, Dr. Doipayan Sinha, to say something about this program. Sir, over to you, please.
Am I audible, Tariq? Hello.
Yes, sir. Yes, sir. Perfectly audible.
Thank you, Tariq, for handing over me the speech. I heartily welcome all the resource persons, eminent speakers, and the participants to the international webinar in the world of plants, and that is organized by our botany department of Government General Degree College. First of all, I would wish to thank Professor Sheikh Bakhtiaruddin of University of Chittagong for being kind enough to join the inaugural session. I also wish to thank Madam Shana Chaudhary, who's also from Bangladesh. He's also there in the inaugural session. I also heartily welcome Professor Devdulal Banerjee, who is the special guest of the inaugural session, and Dr. Subrata Raha, who is also the special guest of the inaugural session. I welcome you all, and I really hope this next 48 hours would be a very highly scientific interaction between all of us, and we are having a lineup of very eminent resource person who would enrich us with their presentations and with their knowledge from the botanical point of view. Now, I would like to request our chief patron and the officer in charge to kindly deliver his introductory lecture, and I would like to request Professor Tariq Ali also to hand over the session to Professor Nimai Chand Masanto, who is the chief patron and officer in charge. Over to Tariq Ali, please.
Thank you, Dr. Sinha, for these kind words. Now, I would request the chief patron of the program and our honorable OIC sir, Dr. Nimai Chand Masanto, to say something about the program. Sir, please.
Thank Dr. Ali. Very good morning, everybody. On this sweet morning, we all meet in this virtual platform to participate in the international webinar on the world of plants, its impact on the indigenous people, society, and biosphere, organized by Department of Botany with the help of other faculty members of Government General Degree College, Mohanpur, Paschim Medinipur. I like to convey my warm greetings to all participants of this webinar. I welcome you all heartily on behalf of this government college as well as myself. I like to share some of my experience about this college. Mohanpur Government General Degree College is one of the youngest general degree college established at the remote village of Kharagpur subdivision of Paschim Medinipur district, West Bengal, and it is nearer to the state Odisha. The college had started its academic journey by 2015 with very poor infrastructure, but with devotion to nurture the poor, needy, but thirstful teenagers with this locality. With the limited resource, our faculty members are trying heart and soul to foster the young blood, to make them a real literary scholar and a good citizen of the country. On this 2 days webinar, so many reputed, illustrious and distinguished scientists, educationists and other eminent persons of repute of different fields will deliver their valuable lectures and share their innovative ideas among the participants. We all shall be enriched with knowledge about our native plants, that is plants indigenous to a given area in geologic time. These plants are native to a particular area. We have many indigenous species having unique properties used to develop specialized drugs, which is our lifesaver. They help to reduce air pollution, provide shelter and food for wildlife. Birds, the inhabitant of the native plants, acts as a pest controller, while the garden provides them with feeding, breeding, nesting, and roosting. Indigenous plants promote biodiversity and stewardship, that is taking care of our natural heritage and ecosystem. Indigenous plants are so important to native people for their nutrition, day-to-day foods, drugs, shelter, and many other reasons also. So we have to acquire knowledge much more about these plants to shape ourselves, our life. If our natural resources are lost, we are going to be threatened. So the topic of this webinar is very much important for our society, especially during this grave pandemic situation. So last but not the least, I again convey my heartfelt gratitude and a profound appreciation for all participants. I am thankful to our eminent resource persons who will deliver their valuable ideas in this virtual platform. Wishing a grand success of this webinar and pray to God so that all of us can maintain our safe and healthy life and stay out of harm's way. Thanks you all.
Thank you, Dr. Masanto, for your inspiring words. Now, we feel privileged that we have resource persons and audience from different parts of the country and from our nearing country as well. We especially feel ourselves privileged to have with us in this session Dr. Sheikh Bokhtiaruddin University. Sir, I'd like to request you to say something about our initiative, if you kindly unmute and say something about this initiative that we have taken. Dr.
Good morning, everyone. Can you hear me?
Yes, sir. Yes, sir. Yes, sir.
Can you hear me? Because I have problem with the network. I have no electricity. I'm going on that.
Hello, sir. Bokhtiaruddin has left the meeting because of some network issue, I think. Vipan, sir. have our resource person of the first session joined?
Yeah, Dr. Raha is supposed to join within a few minutes. I think Professor Devdulal Banerjee is there with us. Sir, are you here? Are you there? Hello? Hello? I think I need to contact.
Yes, yes.
Okay. Professor Devdulal Banerjee is there. Tariq, can you kindly initiate the program now? And one minute, please. I would also like to welcome one of my teachers who have just joined, Professor S.R. Yadav from the Shivaji University, Kolhapur. Welcome you, sir, in this event. And now I hand over the a warm welcome you, sir, and welcome Professor Devdulal Banerjee to this event. And Tariq, please initiate the session.
Okay, okay, sir. So before we start the technical sessions, we have a special session before all this. Now, in this special session, we have two speakers. Professor Devdulal Banerjee, Professor and Head, Department of Botany and Forestry, Vidyasagar University, and Dr. Subrato Raha, Head, Department of Botany, Sidhu Kanu Birsa University, Purulia. This special session will be chaired by our Honorable Officer in Charge, sir, Dr. Nimai Masanto, sir. So I would request, this session actually will run till 10:25. Now I would like to request Dr. Masanto to take the charge of the session. Over to Dr. Masanto. Sir, you are not audible. Please unmute yourself. Sir, am I audible, sir? Sir, but your voice is not coming because you have to unmute yourself, sir. Yes, now.
Am I audible?
Yes, yes, now it's clear.
It's my great privilege to introduce Professor Devdulal Banerjee. Professor Banerjee presently serving as Head, Department of Botany and Forestry, Vidyasagar University. He did his postdoctoral work under Professor Gary Strobel at Montana State University, USA, as a Boys Cast Fellow in Endophytes Biology. He is the recipient of award like gold medal in both UG and PG, ceremony gold medal, CSIR NET with SP Mukherjee Fellowship, Asian Molecular Biology Organization Fellowship, to attend a training on molecular biology at Tokyo, Japan, DST Fellowship to attend a meeting of Nobel laureates and students at Lindau, Germany, DST International Travel Support to attend International Congress on Plant Pathology at Turin, Italy, Boyscast Fellowship to do advanced research at Montana State University, USA. Shikha Ratna Award 2020 from Department of Higher Education, Government of West Bengal. He visited countries like Italy, Germany, USA, South Korea, Japan, UAE, etc. in different academic assignments. Published more than 60 papers in International Journal of Repute, and total impact factor of publications are more than 40. Presently, H index of publication is 25 and I10 index is 40. Published more than 6 books chapter in Springer and CRC Press publication, and a book from Vidyasagar University Publication Division. Submitted more than 25 sequences in Gene Bank. Completed 4 research projects as principal investigator. Currently serving as editorial board member in Microbiology Insight, Journal of Advanced Microbiology, Journal of Pure and Applied Microbiology, Frontiers in Microbiology, etc. 10 candidates got PhD degree under supervision and 8 working in different fields of microbiology and plant microbiome interaction. Professor Banerjee will deliver a lecture today titled plant microbiome and its importance in human welfare. Professor Banerjee, please. So over to Professor Banerjee.
Thank you, Dr. Masanto, for kind introduction. I also thank Doi Payon Sinha and other faculty members of the department and all the organizers. They really did a hard work to organize such an international event. We have with us several resource persons from India as well as from Bangladesh. I must thank all of them. Now I'm actually showing my presentation. Is it visible?
Yes, sir, it's visible.
Yes, sir, visible. Please go on.
So I'm going to deliver my talk on plant microbiome and its importance in human welfare. So first of all, what are different plant microbiomes? In which part of the plants they are actually available? If we consider three different zones of the plants, actually the number of microbes is higher on those particular regions. That is, first one is the rhizosphere. It is the zone between the root and the soil. So it's a particular type of environment where the microflora is quite different from other parts of the soil because from plant tissues some lichid comes out and which attracts the microbes. Somehow influences the plant growth also. They sometimes helps the plants to survive. And second one is endosphere. That means throughout the plant there we can found some types of microorganisms. Maybe they may be beneficial or sometimes they may be pathogenic, but somehow they remain within the plant. And third one is the phyllosphere. That is the leaf surface, because we know that leaf surface contains some cuticular layer around its surface, but sometimes actually cracks are formed in these surfaces, and through which some leachate comes out from the leaf tissues, and that actually attracts different microorganisms, so that the microflora, a unique microflora is formed within these leaf tissues. And sometimes it was found that this leaf microflora, they also influences plant growth. Sometimes even some nitrogen fixing microorganisms were found on the, those particular sites. So these are three different zones. Still, I am concentrating my my talk on the second one, that is on endosphere. So how plants microbe interaction actually influences plants. Here I have shown that they help in stress adaptation. They sometimes act as biofertilizer, biopesticide. In below ground interaction also, the microbes have some role. They have some role on in different environmental interactions. So in different ways, these microorganisms, they help the plants and they increases their survival capacity also. So among these microorganisms which remains there in the plants, they are actually called as the endophytes. And these endophytes are actually fungi or bacteria which all are part of their life cycle invade the tissues of living plants and cause unapparent and symptomatic infection entirely within the plant tissues, but causes no symptoms of disease. So whenever we observe any symptom within the plant because of infection by any bacteria or fungi, that is that microorganism is called as pathogenic. But whenever they remain within the plant, but we are not observing their appearance, any symptom from outside, they are called as endophytes. And these are the microorganisms. They are actually found in almost every plant on this earth. So microbe and plant relationship through this endophytic relationship, that is everywhere, uh, from lower group of plants to higher group of plants. Everywhere we can find such type of interactions, and in some cases their presence is very much essential. Otherwise, in absence of them, sometimes some plant cannot survive also. And these organisms reside within the living tissues of the host plant, and they do show in a variety of relationship, ranging from symbiotic to pathogenic. Why we are telling pathogenic? We are telling endophytes still pathogenic. Sometimes some people, they say is that these endophytes, they are the latent pathogens. That means whenever they uh, uh, they get weak, actually plants immune system when becomes weak, then sometimes these endophytes may express themselves as pathogens. Therefore, they are sometimes called as latent pathogens. And these endophytes, they may contribute to their host plant by producing different substances. They provide protection, and ultimately they help in the survival values of the plants. Uh, Endophytes, they also help in phytoremediation. We know that plants sometimes they remediate different types of toxicants present in the soil, and uh, part of that remediation process is contributed by plants, but the endophytes present within them may also have the role in degradation of those components, or volatilization, or removal in different processes. They actually takes part. And this is the zone of interaction within the root tissue. If you see this root region, the apical cap and the meristematic tissue, and in this particular region, the tissue is so soft that any microorganism may penetrate the root cells. So through this region, actually the endophytic microorganisms sometimes they invade from soil to the root zone, and through this they actually goes through this particular region, that is the root hair region, and through these root hairs they may comes out again in the soil environment. So this is the process. This is called as rhizophagy cycle. In this cycle, actually the endophytes they penetrate through the root tissues, because somehow these endophytes they not remain during seedling formation. May or may not be some of the endophytes are there within the plant or not, but sometimes from soil environment some of the endophytes they actually enters into the plants. So it is one of the possible theory that in this way the endophytes they penetrates the plant tissues. And this is one of the example that what we have found, that the bacteria actually they penetrates in this tiny root region, the root hair cells, where you can find that several bacterial cells are there. So this cloud of bacteria within the root tissue indicates that they are not doing anything wrong within those tissues, and uh, there is a actually mutualistic relationship among this bacteria and the plant cell. Now, the application of these endophytes, actually people they are in search of the microorganisms which have some biotechnological potential. So considering that, already we know that the uh, soil environment already being explored for different microorganisms for production of different components. So people are in search of a new niche where from they will get some unique microorganism. Those can contribute in uh, different product formation, and for that they actually select the plants from these they actually uses these three approaches by which they actually select the plants. And first one is the plants from a distinct environmental setting have the possibility that that plants may possess some unique microorganism with that particular plant. Plants with ethnobotanical history may have the chance to give some unique endophytic microorganism, and some plants, those which are producing bioactive natural products, then from that particular plants also chances of getting some unique microorganism that have the ability to produce those kind of natural products, product formation will be there. So these are some unique environment. We know that giant baobab tree. These are actually found in Madagascar region of Africa, where this particular plant it has some unique features, and they are surviving. Their huge plant growth is found in that dry territory. So how they survive there? uh, or the spiny forest in southern Madagascar, or this Carao River plant. What we are getting this particular plant is remain there within the aquatic environment, and there is a huge water current. Still, they are surviving there. That means they are they are surviving on that particular environment. Maybe some endophytes are there within those plants. They are giving them some survival benefits. So that's the one approach. And another approach I told you a few minutes before, that some plants, if they are producing some particular chemical, so chances will be there that that particular plant may harbor some endophyte which may produce that particular component also. Example is, we know that Taxol, one of the most important anti-cancer agent, and this Taxol is produced from and this Taxus brevifolia, it has a particular habitat. It is generally found in Indo-Tibetan plateau. This particular plant, from this particular plant, whenever we have collected this Taxol, then actually the amount of Taxol production was limited, so that thousands of plants were actually cut down to get that particular bioactive component. So scientists think that if we are able to find out any microorganism there within this Taxus plant, chances will be there that that microorganism may produce this Taxol, and they are successful. They found one of the fungus. Here you can see that particular fungus, that is Taxomyces andriani. This Taxomyces andriani, when it was cultured in liquid medium, within 7 days of culture, it is able to produce that Taxol. And that after the recovery of Taxol from this particular fungus, actually a new way of research, it actually comes out. Till date, what are different products being reported from endophytic microorganisms? These are some of the components. they produce antibiotics. Their bioactive components are being used as antibiotics, antiviral components, and then volatile antibiotics. I will discuss later about this volatile antibiotic. Then endophytic fungi, they produces anti-cancer agent, they produces antioxidants, they produces insecticidal components. All these are being reported, and they are now some of these components are being exploited commercially also. They are producing anti-diabetic agents, they are producing immunosuppressive components, and the latest one is the production of diesel component. So one by one, I am giving these examples. This is ucidin A and chlorinated macrocyclic lactone. This is actually the product of a bacteria, Serratia marcescens. It was actually isolated from an aquatic plant from that southwest Venezuela. In this particular harassed aquatic environment, how that particular plant survives, somehow the endophytes present within that particular plant, they are contributing chances of production of some wounds are there in that particular plant, and during wound formation, infection on that particular wounds will be there. But this ucidin A, these actually helping that particular plant to survive from those pathogenic microorganisms. Then it is another component, the cryptocandin A, antifungal peptide. It was actually isolated from Cryptosporiopsis squarciana, an imperfect stage of Pagicula, you all know. And this fungus was actually isolated from a hardwood species from Europe. The plant was Tripterygium olfori. Actually, from this particular plant, this antifungal peptide was isolated. Then another one is ambuic acid. This is a cyclohexanone produced by Pestalotiopsis microspora, and it was also isolated from a rainforest plant. And this ambuic acid, it is actually used as an antiviral agent. Then from Fusarium, that Fusarium was isolated from Selaginella, and it was the plant actually, Selaginella was isolated from Costa Rica, which produces a particular antifungal agent CR377, which is active against Candida albicans. Then plants like Artemisia annua, which produces anti-malarial compound, and it was found that endophyte present within them also produces such type of components. Then another Streptomyces species, it was actually isolated from Grevillea tree, which produces cacadiomycin, a specific type of antibiotic. This is the Grevillea plant from which this particular Streptomyces was actually isolated, and which produces this antibiotic cacadiomycin. Another antiviral component that is cytonic acid A and cytonic acid B. These were actually isolated from a endophytic fungus, cytonemia. Then about volatile antibiotics. Actually, antibiotics, whenever we take those antibiotics, it enters into our body and it kills the target microorganism, and after that, in some way through excretory process, they goes out. But they have some side effects. So this particular type of antibiotic, that is volatile antibiotic, it was the discovery of Professor Gary Strobel. He was found a particular type of fungus. When he kept that particular fungus in a box containing other fungus also in petri dish, actually there were several petri dishes of different cultures, and he kept all those petri plates in a box. And after 2 days, what he found that excepting a particular fungus, all other petri dishes containing fungus were dead. Why that thing happens? Why this particular fungus is surviving there and all others are dead? So he repeated that thing, and he found that excepting that particular fungus, all are dead. And he actually taken that fungus under microscope, but he found that it's a sterile fungus. He named that fungus as muscode or albas, and that particular muscode or albas, it is a member of Xylariaceae. Actually, he isolated this particular fungus from Cinnamomum gilaricum, from cinnamon tree. He actually isolated that one. And this particular fungi, when it grows, it produces different esters and acids which are antimicrobial in nature. And as because these are esters and acids, they after some times, they as their volatile quality is also less, so they goes out from that particular region. So he named these as volatile antibiotic. Endophytes, they produces anti-cancer agent. I have already told you that Taxol was isolated from Taxomyces andriani, this paclitaxel, which was actually collected. This Taxomyces andriani is an endophyte of Taxus brevifolia. Then another one is taurinic acid, a selectively cytotoxic quinone dimer. It was isolated from Pseudomonas microspora, which was originally isolated from Taxomyces taxifolia, another plant. So both of these components are anti-cancer agents. They are also producing antioxidants. Different antioxidant components are reported till date. They are producing insecticidal components also. Here you can see that one of the endophyte noduli sporium, it was from isolated from Bonsia daphnoides. It produces that insect repellent component, and some of these components are also insecticidal. Anti-diabetic component, that is L783281. This particular metabolite was isolated from Pseudomacharias species. Then immunosuppressive components are also reported from different endophytes, and this unique one is the Mycodiesel. It was also the discovery of Gary Strobel, who have collected plants from different parts of the world. Actually, he explored different rainforests, and from those particular rainforest plants, one of the fungus, Geocladium roseum, it's a electron microscopic image, but electron microscopic image generally gives us black and white photographs. Here it is actually falsely colored, but whenever it grows in the petri plate, it actually gives a rosy rose or reddish coloration on the plate. Therefore, it was named as Geocladium roseum. This endophyte was isolated from Eucryphia cordifolia, that produces 15 different volatile hydrocarbons. And what he found, that most of these hydrocarbons are the components of diesel. Though octane is the major component, still other than octane, different components are present in diesel, and there are a large number of components are also produced by this Geocladium roseum. Then he tried to grow this in artificial liquid culture medium, um, uh, he is able to collect those components there. But as the amount of that particular diesel component is less, therefore he was not successful. But one thing that tells us that somehow these microorganisms, they may have some role in diesel formation, because whenever the plants or different other tissues, they are buried within the soil for several thousand years, maybe their the endophytes which remain there within those plant tissues, they may be converting those tissue components into diesel components. So it's a new way of thinking about the diesel formation using fungus. Now about our work, what we are doing in our laboratory. Uh, uh, because my topic was how endophytes, or how plant microbes, they are actually helping human beings. So major focus in our laboratory is on these particular 4 topics, that is volatile antimicrobials, exopolysaccharide production from endophytes, different enzyme production from these endophytes, and antimicrobial component production. This is the basic technique how to isolate endophytes. Just you have to take your plant tissue samples, and you have to immerse that particular tissue sample in 70% ethanol. You keep that tissue for a few minutes, and after that you have to take it out and dry it out. And then we have to cut that particular tissue into small fragments, and we have to place that particular tissue in water agar plate, so that if endophytes are there within that plant tissue, they will come out from that particular tissue within that agar plate, and from that agar plate we can transfer that particular tissue, that fungal or bacterial growth, actually in a new media. Maybe we use PDA for fungal growth, or nutrient agar for bacterial growth. And the technique is like that. Then incubation, then transfer into PDA plate for fungus, and then we have to observe that under microscope what kind of fungus that is, and then preparation of liquid culture for production of a particular bioactive component. So these are some of the unique organisms we have found. From Osimum sanctum, we have isolated some unique fungal strains. We got Alternaria and Penicillium. Then we liquid culture that one. Then another fungus, that is Halopas. Then Fusarium, it was liquid cultures. Sometimes we got some unidentified fungus also. Their reproductive structures are not actually matching with any of the fungus available in the literature. Then we keep them for actually molecular identification. We send them for molecular identification, and after that we generally get their identity. So these are different fungal members. This is the unique fungal member. What I have told you, that Muscodor albas, it was actually isolated by Gary Strobel from cinnamon plant. And when I traveled his lab, he told me that you may get some of the Muscodor albas in your country also. You just try there whether you are getting any Muscodor or not. But you have to collect your plants from the rainforest region. Then what I think that in our country we have, these are the particular areas where we can get rainforest. Then I travel to a particular region, that is Meghalaya, Moulolong village, you all know. From there I have collected this piper plant. Actually, I have collected several hundreds of different plants, and luckily I have isolated one muscodor species. It was named as Mo12. This muscodor species actually it resembles in structure with the earlier isolates. It produces some sterile hypha, and this sterile hypha produces some some aggregate of tissues, some unique tissues. And after molecular identification, what I have found that it is a muscodor species which have close similarity with other muscodor species isolated till date. And this muscular species, it produces a lot of different volatile components. A major component though was ethanol, but it produces ethyl ester of acetic acid, then propanol, then methyl ester of propanoic acid, pentanol. All these different components, these are in aggregate, they are antimicrobial in nature. And this is actually the split plate test. With this split plate test, whenever you place muscodor alvas in one half of the plate, if you grow there, and if you place any pathogenic microorganism in another half of the plate, what you will find that in most of the time, this muscodor alvas, it do not allow those microorganisms to grow there. As there is no direct or physical contact among these microorganisms, that means no component from this muscodor species is diffusing towards those microorganisms, because there is a channel, so no chances will be there that something is diffusing from there. So somehow some volatile components, they are contributing in this process. And our muscodor species, muscodor alvas, it is able to kill most of the plant pathogenic fungal strains. Here different plant pathogens we have used, we have tested in Sclerotonia, Pythium, Geotrichium, Botrytis. It kills almost all of them, excepting Fusarium, Trichoderma, and Aspergillus. In these three cases, it inhibits them, but their inhibition is a percentage-wise. It inhibits 100% Aspergillus in case of Aspergillus, and 40% in case of Fusarium, but it is unable to kill them. That means their growth is somehow suppressed. We have published that one. Then another one, another fungus, this is not muscodor, but this is Dallinia bambusicola. It was actually isolated from another plant from northeast, and this particular fungus was isolated from Camellia caduca. This from this plant we have isolated this one, and during split plate test, what we have found that it is also inhibiting different microorganisms, not killing, but it is also inhibiting different pathogenic microorganisms. And this is the electron microscopic photograph of this Dallinia bambusicola. And when we did gas test for this particular microorganism, what we have found, the different components it is producing are antimicrobial in nature, and among these two components, linalool oxide and linalool, these two components, they have a sweet fragrance. That means whenever we grow that particular fungus, it actually produces a sweet smell there, and so possibilities are there that we may use this microorganism to clean the air as well as to get a good meal also. And we did this particular test. We have placed some of the fruits and vegetables in a cardboard box, and we placed this Dallinia bambusicola culture in a half of this particular half of the box. And whenever in control, when we have not given any Gardenia bambusicola culture in those particular boxes, what we have found, their spoilage uh, uh, happens earlier than the experimental one, where their actually shelf life is extended for few days whenever we use this one. This indicates that actually uh, they may be used in a biofumigation process by which we can increase the shelf life of fruits or vegetables uh, um, actually which is beneficial also, because the components, the volatile components, most of the components, they are not toxic at all. So whenever we use them, and after removal of those things, just if you wash these fruits or vegetables under tap water, this will be safe for human consumption. So this is the actually table which indicates that in most of the cases it is actually killing Phytophthora, this Daldinia, it is killing Phytophthora, but inhibiting all other fungal strain in these percentages. This is saccharide. First, we isolated from one of the fungus Staphylium. We found that this exopolysaccharide is it was a heteropolymer of mannose and glucose. Then another exopolysaccharide we have isolated from Fusarium solani, and this Fusarium was isolated from it was actually isolated from Calmette plant. Its polysaccharide was actually purified. We got two types of polysaccharides, one fraction 1 and fraction 2, and this polysaccharide was also nature of the polysaccharide was also detected with different instrumental techniques. We did paper chromatography, then GLC. With that, what we have found that this is a heteropolymer. And we did transmission electron microscopy of that particular exopolysaccharide. We have found that its fibrillar nature that indicates sometimes polysaccharide they are globular, but here in this case this polysaccharide is fibrillar in nature. We did its different bioactive tests. Here it is the membrane stabilization activity, mast cell protection activity. What we have found in both the cases, it is giving positive result. It is malonyl level. It contributes in membrane stabilization process, or it is in case of peritoneal macrophage actually they plays some role. And what we have found that with the increasing concentration of this exopolysaccharide treatment, malonyl level actually increases. Then another one is superoxide dismutate activity. Then this is the growth curve of that particular fungus. And these are different publications. Another polysaccharide was isolated from another Endophytes. It produces actually it's a homopolymeric sugar polysaccharide. Now another fungus, Hyalopas, it produces tannage, one particular enzyme which actually degrades tannin. It's growth curve. I do not have enough time, therefore I'm just skipping those things. Another actually actinomycetes I have isolated in my lab, that is streptomyces, which produces tannage also. From Astronia scholarlies, we have isolated a lot of different fungal members. They are producing different types of antibiotics. This is a streptomyces. We have isolated it from Cinnamomum thamala, the Tejpata plant, and it produces this particular antibiotic, antinomycin, its chemical nature was actually confirmed through NMR studies. And what we have found that it is active against penicillin-resistant and penicillin-resistant Staphylococcus aureus. Another was isolated from Catharan fastidiosus, this particular Endophytes tectomyces thermoviolaceus. It produces another antibiotic, that is granitinic acid. Another one was isolated from Blumia lacera, Streptomyces riccius. It also produces different antimicrobial components in its culture mixture. So these are the references, and I must acknowledge all these people listed here. So thank you.
Thank you so much, Professor Banerjee, for your voluminous lecture and nice presentation on plant microbiome. We are enriched with your nice lecture. So many thanks to Professor Banerjee. Wishing your safe and healthy life during this pandemic situation. Please be safe, remain healthy. So our next speaker, Professor Subroto Raha. I welcome Professor Raha. It is my pleasure to introduce him. He is the head and Department of Botany, SKBU Purulia. He has teaching experience of more than 25 years. He is an ex-assistant teacher in life science, Ramakrishna Mission Vidyapith Purulia, ex-assistant professor in botany, Raghunathpur College Purulia. He has received many awards like recipient of National Scholarship. He secured second rank in BSc exam. He is the recipient of Bannojiva Dronacharya Award and Porivaram Dronacharya Award. He is the Scientist of the Year 2019 awarded by International Academy of Science and Research. He is also awarded Shiksha Ratna in 2020 by the Higher Education Department, Government of West Bengal. He has membership in different professional bodies like Botanical Society of Bengal, Social, Environmental and Biological Association, Indian Aerobiological Society, International Academy of Science and Research. He presented papers in 43 conference, seminars, symposia. He has delivered invited lectures, acted chairperson of 68 national international conference, seminar, workshop. His publication include 19 abstracts with ISBN number, book chapters and 6 are under press, 22 in journals and 2 under process. He has 2 ongoing research projects and he has completed 1. Today, he will deliver a lecture titled Diversity in Pteridophytes in Purulia district. I welcome Professor Raha. So over to Dr. Raha. Please.
Thank you, sir. Respected delegates, the principal of Government General Degree College, Mohanpur, the organizers, especially my dear brother, Dr. Dwipayan Sinha. I am delivering a lecture on diversity of pteridophytes in Purulia district, particularly with the reference on ethnobotanical importance. I am sharing my slides and it is Is it visible?
Yes, yes, sir.
Yes, sir. Visible,
Yes, sir. Visible, sir.
Okay. So this is our university at Purulia. I welcome all of you to the Shidukanu Birsha University, which have started its from 2010. And botany department has initiated 4 years back from 2016 only. First of all, I want to highlight some of the characteristic features of this district. Regarding the soil, mainly residual soil, which is derived from the granites and mist, is the characteristic soil type. And lateritic soils in the upland area is frequent. Clay loam and clay interspersed with gravels is also another characteristic feature of this region. Soil is mainly acidic, ranging the pH ranges from 5.5 to 7.2. And soil erosion is high and mainly humus accumulated in the valley regions where the soil is much more fertile than the rest. Now, these ferns are of importance in the folklore, and the plants successfully used in different systems of the medicines like Ayurveda, Unani, homeopathy, and other systems of medicines. And the tribal people mainly use the rhizomes, fronds, and spores, and particularly the leaves for various to cure different ailments. And May published a detailed review, where he listed 105 medicinal ferns. In a recent compilation by the Singh, reported 160 species of useful pteridophytes in India on the basis of its phytochemical, pharmacological, and ethnobotanical studies. And it's the antibacterial or antimicrobial property has also been established by different research works. Now, these are the region. Apparently, the rest of the India knows Purulia as a drought-prone region, but the hilly regions are much rich in biodiversity, different ferns, bryophytes, and herbs, shrubs, medicinal plants grows here. In the above you can see this is the Panchet or Panchakot hill, which is a treasure box of different kinds of medicinal plants and orchids. This is Joychandi hill. It harbors many pteridophytes, particularly 2 species are very much abundant here, which I am going to discuss later on. And this is Manbajar region, Jharnakocha, and it is also densely forested. Here, the Ajodha hill ranges at the below, you can see, and this is a party, and these are different falls which runs through the hills. And these are the different documentation sites of different corners of Purulia. And particularly, this is the Purulia town. From a satellite map, you can see that this is the Ajodha hill from where most of the plant species were recorded. Here it is the Hottan forest near Jhalda, where many species has been recorded. I'm sorry. And this is the Panchakot hill or Panchet hill. This is the Panchet reservoir. This is Panchet hill. Here a small patch of forest you can see. This is called Raka forest. This is Bandwan and Manbajar region on the south, where these are the mainly collection sites or documentation sites. You can see in this map also, these are the Ajodha hilly region. This is Hottan forest. This is Panchakot hill on the north. On the east, Rakab forest near Puncha, and here Jharnakocha, Barabajar region. These are rich in different kinds of plant species. Total 35 species of wild pteridophytes belonging to 12 families under 24 genera were recorded. The dominant family was Pteridaceae with 10 species, followed by Salviniaceae, Ophioclausaceae, each with 4 species, Polypodiaceae, Philipteridaceae, and Silaginaceae, each having 3 species. Lygodiaceae and Marceliaceae each represented by two species, and the rest of the families like Atheriaceae, Equisetaceae, Glyciniaceae, and Isoetaceae are represented by only one species. And if we consider their habitat, then mostly we are getting terrestrial habitat. Among them, 54% is terrestrial, epiphytic 9%, aquatic 34%, and semi-aquatic 3% pteridophytes recorded from this region. Now, this is a list of pteridophytes, what I am going on later on, one by one. And this column indicates its nature. That means SA means semi-aquatic, T means terrestrial, A means aquatic, and so on. These are the different species what recorded from this region, and it is communicated for publication. Now, Adiantum incisum, where the leaf powder mixed with butter or ghee, it is mainly the reports, mainly the ethnobotanical informations collected from the Santhal and Bidhot tribes of the hilly region, and also they are very much cooperative, some not very much cooperative, but sometimes due to our creating a rapport with them, we have explored some of the information from them. It is used in cough, fever, and skin diseases, and leaf juice is also applied to check hair fall. Adiantum incisum, and that is, it is common. The next one, which is Adiantum filipens, grows everywhere, where the root decoction is used for chest pain, and rhizome powder is used as an antidote for snake bite. Leaf juice used in dysentery and burning. Anuretopteris bicolor is another interesting silver fern, commonly called. The rhizomes, type, and lamina is antipyretic, antiseptic, and the mature rach is used as ear and nose studs for girls. That is, after puncturing the nose and ear, generally we use the rachis of the neem, but the tribals use the rachis of this plant as a stud after piercing their nose and ear for ornament, for wearing nose ring or ear ring. Azolla pilata is common, but no ethnobotanical data has been recorded for this species. Ceratopteris thalictroides, here you can see a large field is covered by Ceratopteris thalictroides in Balrampur region. Fertile shoot and the sterile shoot are different. It is used as poultice in skin diseases, prawns, and wounds. This is another silver fern, Chailanthus tinifolia, but no ethnobotanical information has yet been recorded. Priscilla dentata is also common in the plainlands, and its antibacterial activity has already proved, and the whole plant is a good cattle feed. Dicranopteris linearis also occur in the certain parts of Bankura and Midnapur district. Dicranopteris occur certain parts of the Garbhata region of the I have observed, and elsewhere also. And here it exhibits a luxurious growth, and leaf and rhizome extract used in burn, itching, and rash, particularly skin rash. Cyclosorus pteridophyte used to treat shores. This is Diplasium esculentum, where as insect and pest repellent, rhizome extract used to treat spermatoria. T6 uses vegetables. Some years ago, the Ajanta Hill region was disturbed due to some agitation, and at that time, several Naga forces settled here, and they frequently collect this plant and use as vegetable. We have observed. So this is Diplasium esculentum. This is Helminthos is Gilanica, occur in the very dark and humus-rich region, particularly at the valley regions of the hill near Bagmundi. Leaf extract used in cough, tonic, and to cure the sore on the tongue. Next, recently we have identified or documented Hemionitis erifolia, which is very interesting fund, and the whole plant and frond is considered as hypoglycemic. It is already documented, and particularly it is used as anti-diabetic and to remove intestinal worm. And here near a village called Shonahara in Ajodha Hill, particularly in a very shady place, we have isolated this, we have documented particular Hemionitis erifolia. Equisetum only observed in 2 pockets on the Ajodha Hill, and it is Equisetum ramosimum and subspecies with subspecies debile. Here the rhizome extract used as tonic by the tribal people and only present till now we have observed in 2 pockets on the Ajodha Hill. This is Isoetes coromandeliana, where the whole plant gives out a fluid used in the treatment of liver and spleen diseases. It is an aquatic pteridophyte. 2 species of Lygodium recorded. Lygodium flexuosum is given here, and Lygodium japonicum is believed to be an escape variety. So here I didn't mention it. Only Lygodium flexuosum, where the plant rhizome used as an expectorant by the tribal people and used to treat carbuncle, ulcer, eczema, and coughs. Aqueous rhizome extract is also used to treatment gonorrhea. 2 species of Marsillia recorded, Marsillia minuta and Marsillia quadrifolia. Marsillia quadrifolia with its large leaf and very common, known as Shushnisha, famous for its production of marcellin. And here is Ophioglossum gramineum, very small grass-like leaves, which is used as astringent, and this is also used in wounds. The decoction also used in angina. This is another species of Ophioglossum reticulatum. The shoot is used as vegetable, and the whole plant paste to treat skin diseases and menstrual disorder. This is somehow larger species than gramineum. And this is Ophioglossum costatum. No medicinal importance has been recorded. This is another kind of silver fern, Ptyrogramma calomilanus. And here it is used in renal disorder, fever, and cough. The mature rachis, it is also used in ear and nose starts like Aleuridopteris. And here you can see in the next photograph, Purulia is barren rocky area and how the ferns are growing on this sandstone. You can see this is very dry sandstone, and but Ptyrogramma is frequently growing here, particularly in the Burguma region. This is common Paris vitata. It is juice is used by them as a diarrhea to cure diarrhea and dysentery. This is Pyrotia lanceolata, where leaf extract, it is an epiphytic fund. The leaf extract used in cuts and wounds. You can see the sorus on the reverse of it. Selaginella bryopteris is the characteristic feature of this region. All the more or less, at least 80% rocks in the plain land in hills of Purulia district are covered by this resurrection plant, Selaginella bryopteris, which is diuretic and used to treat gonorrhea and also used to treat gastric disorders. This is see how it is infested over a rock, barren rock. This is another species of Selaginella ripenda, mainly grow in the higher altitude of the Ajanta hills, not occur in the plainlands, and the whole plant used to treat pus formation of ear and for gastric disorder. And another species is Selaginella ciliaris. This is an enlarged view of the Selaginella ciliaris. Two species of Salvinia triculata, which is very common, and Salvinia molesta, recorded from this region, which is proved to be antibacterial and anti-cancer agent. Now, in the discussion, just what I can say that much more care should be taken to preserve and conserve such fern and fern allies in this region. We can construct, we have a plan to construct a fernarium in our university campus, as well as due to different developmental activities like road construction and construction of buildings in the hill region for tourism. Several sites are depleted, and sometimes the Ophiuplosum, which grows on the periphery of the pond in the moist land due to excavation of the plants, due to its increase in surface area, we generally practice excavation and which sometimes destroy the Ophiuplalia. I have observed here in some villages, and so much more practice should be, much more, awareness is essential to preserve these kinds, and their medicinal importance needs to be established scientifically. And these are some references. And I am thankful to my retired professor Radhanath Mukhopadhyay of Bardwan University, who always encouraged me to work in endophytes, and still now I am in good touch with him, and he is inspiring me till date. And I am also thankful to my two students who are standing here. One student is standing here and one is sitting here, Ashit Mahato and Monoranjan Pramanik. Always and every time I get their cooperation in every trip, and for the indigenous people, in the collection of the indigenous knowledge. I am thankful to the different Shantal and Bighod families of Panchakot Hill, Ajanta Hill, and Barawaja region. And thank you. And through this seminar, I also thank the organizer to giving me an opportunity to share our views and reports through this webinar. And I also welcome all of you to the Shidukano Desai University. Thank you very much.
Thank you, Professor Raha, for your nice presentation. Actually, we are living on plants. Your lecture and also the citation of Professor Banerjee enriched us. We are on the plants because, say for food, say for medicine, say for shelter. So each and every step, we have to depend on plants. And very nice after birth, when we take our oxygen, that also we take from plants. That is a gift from plants. So many thanks to all of you. Now I hand over this session to Dr. Doipan Sinha. Dr. Sinha, please.
Thank you.
Hello, sir. Am I audible? Hello.
Yes, you're audible, sir.
Oh, okay. Thank you, Honorable Officer in Charge, the Government General Degree College, Mohanpur, and who is our chief patron, for being kind enough to chair this session. Energy and for their wonderful presentation, and With due permission, if Dr. Mashanto permits the end of the inaugural session, I wish to take his permission to proceed with the next or the first technical session of the day. Sir, Dr. Mashanto, are you available? Hello.
Yeah,
Hello.
I am available. Yes, I am available.
Sir, okay, sir. So, shall we proceed for the next session with your due permission?
You will go on. Please,
Okay.
please proceed.
Thank you, sir. Thank you for giving us the permission to proceed for the next session. And the next session would be. Hello. The next session is our the first technical session, and our first distinguished resource person, Professor S.R. Yadav from Shivaji University. He will be delivering a lecture, and I personally request Dr. Subrata Raha for kindly chair the session of the first technical session. Professor Subrata Raha, are you there? I request you to chair the session, please.
Okay, thank you.
Okay, thank you, sir. Thank you for chairing the session. I once again welcome Professor S.R. Yadav. He's a wonderful person. I am very fortunate to have his blessings. He was my teacher during my post-graduation, and we are very lucky to find you here in the event. Okay, Dr. Subhata Rama. So the session is yours. You kindly please proceed with the session. Thank you, sir, and welcome, Dr. Yadav.
Welcome, Professor Yadav. And we are very much fortunate to get you in this session. Particularly, we have read your several papers. My scholars are very much fond of you, particularly in connection with BSI. So very much thankful that you are present here. Professor S.R. Yadav completed his BSc from Jaswant Rao Chauhan Science College, Karat. MSc and PhD from Mumbai University, Mumbai. He joined JSM College, Alibag, then Waze College, Mulund, and Shivaji University, Kolhapur in 1985. He worked in Goa University from 1992 to 1995 as reader and head in Delhi University from 2002 to 2005 as a professor. He served as professor from 2005 to 2016 in the Department of Botany, Shivaji University, Kolhapur. Then he was awarded UGC BSR Faculty Fellow and worked in same department. Now he is working as INSA Senior Scientist in Shivaji University. He is interested in he is interested in plant exploration of Western Ghat, biosystematics and conservation of rare and endangered threatened plant species of Western Ghat. So why he is famous to us? He has guided 11 students for MPhil and 26 students for PhD. Along with his students, he has published 8 books like Flora of Kolhapur District, Know Your Grass Genera Through Handlands, Grasses of Maharashtra, and Flowering Plants of Western Ghats. He has contributed chapters in more than 10 books. He has several awards, honors. Here I have the honor to say about that. He awarded Best Teacher Award of Shivaji University, late Professor V.V. Sivarajan Gold Medal, late Dr. J.D. Apte State Level Award, Dr. Egbote Prize, ARI. He is the professor. He is awarded Professor Panchanan Maheshwari Medal, Father Santapao Medal, E.K. Janki Ammal Award, Dr. V. Puri Memorial Award, Boiddo Khariwale Samstha Bonamitra Award, and he is the live member of Indian Association for Angiosperm Taxonomy, Bombay Natural History Society, Bombay Phytomorphology and Botanical Society of India and few others. Dr. Yadav is an outstanding plant taxonomist of international repute, an excellent teacher whose floristic and taxonomic works, particularly Flora of Kolhapur district and Grasses of Maharashtra, have tremendously influenced many students to take up plant taxonomy as their career. 6 plant species have been named in his honor. And many more things have to say about him, but due to paucity of the time, I am just concluding now, and he will deliver a lecture on role of educational institutes in documentation, conservation, and sustainable use of plant diversity of India. Sir, Professor Ishar Yadav, please.
Thank you very much, Professor Subrata. Please allow me to share my screen.
Sir, present now?
Yes, I am.
Yes, sir. Yes, sir. Okay, sir.
Sir, allow me to share my screen. Sir, allow me to share my screen.
Sir,
Himanshu, are you there? Please guide Professor Yadav to share his screen. Hello. Hello.
Yeah, Professor Yadav, you're not able to see the sharing option?
Present now, but sharing option is not seen.
yes, sir.
Present now. Once you click on present now, then click on
Sir,
your
after-
entire screen, and then
right,
you
sir.
should be able to do that.
Right.
Yeah. Thank you.
Sir, when you are clicking present now, then
Your entire screen or a window. Two things.
then click on there, sir. Click on that box.
Entire screen would work, sir.
Entire screen.
No, it says you must grant permission in order to screen share.
Abhishek, can you check the permission option for Dr. Yadav?
Why it is happening?
Hello.
Yes, sir.
Yes, you have permissions. You click on present now, okay, and then you need to click on your entire screen.
On that dialog box, sir, you have to click again.
When you click on your entire screen, there will be a dialog box, and then you need to click on OK, selecting entire screen. There will be a drop down that needs to be selected, that which part of the screen you want to like present, and then you need to click on OK.
You're not still getting the option?
No, options I am getting, but it says that you must grant permission in order to screen share.
Is there an option to request for permission?
That I am seeing. Just a minute, sir.
Sure.
No, actually, Himanshu, that is about his permissions to share the screen.
Yeah, got it. In case it is possible, you can share the deck with Dr. Dwaipayan or someone, and they can share the screen. Maybe Dr. Raha.
Yes,
Okay.
yes.
All right. Okay.
The sharing has started.
Yeah. Thank you so much.
Is it okay now? Are you able to see?
Yeah.
Yes, we are able to see your screen.
Good morning to all of you. It is my great pleasure to talk on role of educational institute in documentation and sustainable use of plant diversity. One thing we should see throughout this last COVID-19 period, all academicians of our country, they are organizing webinars and trying to educate society, trying to educate people, trying to give ideas. What I want to say that we all professors and researchers, students, we are the community which is directly concerned with welfare of human race, welfare of Mother Earth, and what is their role in this world. And I find that the topmost in shaping the world is by educational institutes and research institute. Now you see, I will be talking on plants. Of course, plants, all of you know that any plant, when you see, just don't say that this is a Mangifera indica. It has several other implications which we try to understand. Reason is, every species is a unique creation of nature. It is unparalleled chemist. Just today we heard endophytes, and they produce so many compounds, you know. So they are unparalleled chemists of the world. They are witness of evolutionary history of life. They have great phytogeographical significance, different functions in ecosystem, and range of tolerance and distribution, spatial relationship, and phylogenetic relationship. Really speaking, what is the real capital of human being? Please try, think over it. I have come to the conclusion, it is not money, it is not bungalow, it is a plant or biological materials or biodiversity is a real capital of human being. Every pleasure of our life we derive from this biological capital. So there is a need for serious field surveys, careful observations, critical analysis of various implications of every species. Now you see, educational institutes shape the man and society. This is my sentence, which I say, our primary schools give shape to the thinking of our young children. Then secondary schools, they educate students in various branches, introduce. Our colleges and universities, they really train manpower which works for the country. And then only, you know, we talk a lot about conservation, lot about destruction of forest and so on things, but whole thing starts right from primary school and goes to the research institutes. So we are world makers, and therefore educational institute have a great role to play in sciences. Now you see, just now our professor Subrata said, and our other people also said about plants. Now first slide, you can see this one, no? See this food plate, that comes only from plants. Only plants are able to synthesize food which we can eat. No human beings could synthesize their own food with chemicals. So plants are the probably best scientists of the world. Now this is my stomach. Unless my stomach is full, entire world is useless for me. Then another thing is, you know, these medicines. Just now we heard about medicines on endophytes and pteridophytes, ethnobotanical studies, traditional knowledge. Now, since our origin, whatever diseases we come across or whatever we suffered, plants have come to our health. So my health is also my plants. And third, somebody said about oxygen. See, even if we want to enjoy this entire world, we need to breathe oxygen. And that oxygen also comes from biodiversity, especially plants. And therefore, my breath is also my plants. So my stomach, my health, and my breath, All these things come from plants, and fourth important thing which we need is the water. Soil is important, but these four things seems to be of topmost interest, and our existence, the entire animal kingdom existence depends on plants. And that is what I want to say initially. Educational institute and biodiversity. Now, all over the world, we are facing the great danger of loss of biodiversity. Already we have entered into the into phase of mass extinction. We are losing species in masses. And there, what is important? So first thing which we do is a documentation. Now documentation is it a simple thing? Professor Raha, he documented all the pteridophytes of that district or state, and that is a document, a basis on the basis of which we work further for conservation, for utilization, or for sustainable development. That documentation is done by educational and research institute of the country. So basic knowledge, basic data comes from our academicians, teachers, students, and research scholars. Now, when we come to bioprospecting, when we bioprospect anything, who works for it? Bioprospecting means making plants useful to human beings. Now, our research scholars, our students work in laboratories and do phytochemical analysis, do domesticate or do hybridizations and bring new sources for human beings. Again, here educational institutes play important role. Then sustainable use. Now, who educates society for sustainable use? Again, it is the educational society. And last one, most important, conservation. Who can do conservation? Somebody will say there is a Ministry of Environment and Forestry. They have strength, but they don't have, technical knowledge which they need. Forest department can protect forest, but cannot conserve. Protection is different from conservation. We know red species, endemic species, where they grow, how much quantity, all that data is created by my academicians, by my teachers and students. Now, what is the role of educational institute? Train and introduce biodiversity, train students in identification, documentation and biodiversity, creating awareness about biodiversity. Now, whatever seminars, webinars going throughout country, if you see those seminars, most of the lectures are they are creating awareness about biodiversity and their importance. Research scholars, biodiversity and required knowledge. Research scholar day and night, they are working in laboratories, creating new knowledge, which is used by society. Research projects, which are, through research projects, scholars are, documenting, conserving our biodiversity. And our educational institute are mainly concerned with creation of trained manpower for the task of biodiversity documentation and conservation. Now you see our biodiversity, plant biodiversity, diversity of plants. I will talk about plants because I work in flowering plants, but for me, right from bacteria, viruses till angiosperms, everything is equally important. Viruses, bacteria, this data I will not tell, but still angiosperms, we have more than 18,500 species of flowering plants. It is one of the mega diverse country of the world. 37% flora of India is of angiosperms. Now, I want to tell you that real pleasure of life comes from plants. Now you see these flowers. Each flower is constructed so differently, so uniquely, that to understand single flower, even one life is not sufficient. So biology is not like other subject. It is a very, very different subject. Okay, all these, just I am showing you diversity from Western Ghats. The entire India, if you consider, few slides I will be showing, and I will go to my, see these flowers. Now, this is Anonasi. So much diversity of flowers in Anonasi. Just a few members I am showing. See the designs, see the architecture, see the shape, see the color. Now you tell me, such a diversity, I don't know why people don't get excited with this. We get excited with aeroplanes, we get excited with many other things, man-made things, really excite me so much that I become a lover of plants on this one. Now tell me, architecture of this flower, why nature has designed these flowers? Evolution is not haphazard, it is directional. And you can see this diversity of just Anonaceae I showed. See the flower, each flower has different color, shape, size, and so on. And that way we are lucky that we are Indians, and India is one of the country which has richest biodiversity, right? So such a biodiversity exists. Now see this barlerias, our barlerias, see the colors, shape, size, plants, their growth forms, all these are wonderful. Then these arides, only few I am showing from Western Ghats. Now see this aquatic plant, Eriocolon, Cetaceum. Now see this, entire plant is in leaves, in water, and you can see the rosette of leaves and these white inflorescences which come up. I don't know, such exciting things are there in plants. Now you see, this is one species, Irenocarpus pneumoni. I said in the beginning, different implications of the species. Now this is a monotypic endemic tree species of Western Ghats. It has no close relative in India or in the world, so it is monotypic. How much we know about this species? See, our efforts we are doing, but still we need many more efforts. This is Blepharistemma serratum, another monotypic endemic tree species, which is a rhizosphoraceae member but grows in evergreen forest. What I want to say, now these are the plants, only I have shown two, there is a long list, which are critically endangered, which are restricted to very small areas, and they are they are narrow endemics in Western Ghats. Now, question comes, whether our politicians knows, whether our society knows, only academicians, only research scholars, only researchers, they know about these plants, and their conservation is our responsibility. Why I say educational institutes are of prime importance today in biodiversity conservation and utilization. Now you see one student who worked for 2-3 years in one area, and from an area of about say 20 square meters, he discovered how many species. See, right? So I am just showing, I will not name, but see the species which are described from a single pond in the last 2-3 years. 5-6 species have been described. What I want to say, taxonomy. I mean, there are people who say that taxonomy is a dead subject. Taxonomy has no importance. But, you know, taxonomists have documented biodiversity of the world. And that is the basis for every progress. And therefore, I say that academicians, academic institutes have a prime role. And this is one example I am showing. Now, first question, bioprospecting of biodiversity, who does it? Okay. Now, domestication of ornamental plants, who does? I know NBRI and our tropical botanical garden and many other gardens, they are bringing wild plants and growing in gardens and trying to domesticate them as ornamentals. Screening for phytochemicals or biomolecules, our phytochemist, our botany student, chemist student, they are working on phytochemistry of many plant species which are of great potential. Wild relatives of cultivated crops, today we will be facing problem of food shortage, and there our wild relatives will come to our help in improving our crops, which will cope up with a new environment. So that also work is done by academicians. New resources from plant diversity. Now new resources in the form of gums, resins and whatnot, they also, that research is done by students. Now, you see one example, bioprospecting. Now this is the one species which we described, Crinum mulbaricum. And you know, this Crinum Crinum mulbaricum. It is species just described in 2013 or 14, which has longest leaves in genus Crinum. Leaves grow approximately 5 centimeter in length per day. It is restricted to 0.5 square kilometer in the entire world. You see, 0.5 square kilometers in the entire world, that too small streams. It may not take even 1 month for its loss, and whose responsibility it is to conserve. Now, this is a species of, it is related to Thyanum, it is a tetraploid, and read the last line. It has the highest galanthamine content in the bulbs. So our biochemist student, they analyzed and showed that galanthamine, which is used in Alzheimer, it is a best source of highest galanthamine. What I want to, bioprospecting is done by our students, done by our research scholars. What needs to be learned from this discovery? You just imagine. Now, another thing, wild relatives. Now, you know, Vigna, Moog, Matki, Udid, we call it as. So these are most important crops of our country, and there are about 30 other species of Vigna in our country. How many of we know? How many are there in our germplasm bank? Because these are the plants which are going to come to our help in improvement of our moong dal, improvement of our black gram, and so on. So these are some species which I want to tell you that this exploration is done by botanists. Now, you know, new resources. Today, fodder is a major problem. Of course, human beings talk about only about their interests, but our animals which need lot of fodder, our forest needs grasslands, and we need people who have knowledge of grasslands, grasses, their fodder value. All that research is again done by educational institute. So new resources in the form of fodder, fuel, gums, resins, medicines, etc. is created by our educational institutes, and that will help to feed our animal stock, to conserve our wild animals, because grasslands are very important to maintain wildlife. So, educational institute, I say, it is a heart of knowledge. I mean, we all work for it, pumping this knowledge in the society, and that way I consider educational institute as a heart of knowledge. So, first thing, we do some academics. It is also equally important. Then we educate society, our students. We do research. We try to give ways of utilization. We create awareness. We create and we conserve biodiversity. So how our educational institutes have a great role to play today. This is what I want to tell to my students and my teacher friends. Now you see, this is a one plant species. All botanists of our country should know it is a Trithuria concanensis. Once it was kept near Gramini, now it is basal angiosperms. Can you imagine? This is a sister clade of Nymphaceae. Okay, this is academics. Now this plant which grows in, which is reported from our country, is a Trithuria concanensis, and there is a family Hydatilaceae. And you know, according to APG4 system of classification, every flora in India will start with this family. This is Hydatilaceae because it is a basal angiosperm. This is academics which we do, which has a great significance and satisfaction for a researcher. So this is one plant. All of you should search in the field for its location. Now, then you see, number of student, teachers and research scholars taking guidance in lead botanical garden of Shivaji University. But this is true with every university, every college where we train our students, creating awareness in young minds, and that way training our students. So education is another important aspect of our educational institute. Now, you know, research, student in the field, these are my students who are sitting and analyzing plant in the field. So universities have great strength of talented and hardworking, enthusiastic and passionate students which do research and bring new resources for plants. Then see all these researches which all of you are doing, and this forest department, let us consider forest department, whether they know what are the endemic plants, whether they know where they grow, whether they know how to grow them. So all that knowledge is only with academic institutes, only with educational institute, and then we have to go and train our forest officers, forest guards, what are those species, how to conserve, what to conserve, and where to conserve. All that comes from our educational institutes. You all are playing important role in development of our country. So these are the questions: what to conserve, why to conserve, when to conserve, where to conserve, how to conserve. All these are the questions which you and me, I mean to say, students, research scholars, and teachers have to answer today. Now you see, this is a list of some of the rare, endangered, and threatened species of Western Ghats. Now, conservation is an important aspect for all of us. Now, who can conserve? Forest. Forest can protect. Protected area. They will not allow anyone to go in, collect any plant. But that is a protection. What is a conservation? And for what? For conservation, what you need? You need to know the plant. You need to know when it produces fruits and seeds, whether these seeds are viable, how to germinate seeds, and how to raise seedlings, where to plant them, what is ecology, what is the need of the species, ecological niche of the species. All that technical knowledge is with academicians, is with students and research scholars. Again, I am tracing that we have to help our forest office for conservation of species, and our role is very important. Now you see, just we have done, I told domestication of ornamental plants. In India, there are 18,500 flowering plants alone. How many of them we have grown as ornamentals? Because we are so rich, we don't care, I have 100 of shirts, so I don't care for many of the shirts. So same way in India, we have so many species, but we have not domesticated them. Again, academic institute, horticultural institutes have to play a great role to bring wild species under cultivation as ornamentals. This is one experiment. This is Delphinium mulbaricum, which we did and released as a floricultural crop for our farmers. So such kind of works also are done by. Now, conservation is a most important aspect which I told you. And today, once you all know, if we lose one species, it is lost forever. We cannot create that species by any amount of equipment and any amount of laboratories. And therefore, conservation is the only way to protect these plants or those genomes. Now, this is Hubaridae heptaneuron, this is Ceropegia fantastica, which are critically endangered, and our funding agencies are helping all the colleges, universities for conservation. So this was a DBT project. I thank DBT. And you see, our students with life risk go to the field and do reintroduction, restoration of critically endangered species of our Western Ghats. Many professors in Northeast India, in Western Ghats, they are doing this. Again, this work is not a forest department. It is in collaboration with forest department, but all data is created by we have created. Now, see this conservation. Just these Ceropegias. Who knows these Ceropegias? Again, we know. We botanists, we know. So these Ceropegias, who will conserve? Our ministry can give us funding. Our leaders can support us, but we have to do the work, and therefore educational institute. Our students have done biotechnological tools, conventional tools, and they have restored many species of critically endangered species. They are doing it. Again here, research institute and educational institute have major role in conservation. So just this is Abitulan Ranadi. You can see this one girl has raised thousands of seedling, which is a critically endangered plant and reintroduced in forest department. Now this all, because of time constraint, I am going fast, but slides are self-explanatory that student is our strength, and these students can change the country. They can take up, they can do wonderful contributions to the development of the country. So this way, simple experiment every university should do, that collect the seeds and raise the seedlings and distribute to the people. And this way, Shivaji University has done, and thousands of seedlings have been distributed to municipal corporations, Gram Panchayat, and so on. So every university can do it if they wish. So you can see this, it is not talk, but this is a practical. Anybody can come to Shivaji University and see Lead Botanical Garden. At any point of time, you will get more than 1 lakh seedlings in our nurseries of Shivaji University. So every university has land and they can utilize. Palms, you see so many palms. Last 35 years, I collected palms. Today we have more than 100 species of palms in Lead Botanical Garden of Shivaji University. And all these, Malis, teachers, students, research scholars, all they work for the botanical garden. Now you see this experiment. These are PhD scholars, and I have a lot of hopes from my PhD scholars who can do wonders. There are 2 species which are critically endangered, Camptorrhiza indica and our Dipcady conconensis. In one day, here that ladies hostel was to come up, and they have digged up 20,000 of these plants and replanted at various places. Now, the same critically endangered plant species, they are growing at 10 places, right? So this is the way they have done without money, not that. So I appreciate and put their efforts on record, because I am the only witness for their contribution. We don't do paperbaji, plant one tree and do and give in paper and forget about it. Really, if you want to do work, our research scholar can do. All of you, I hope whole of the India will do it. And see, awareness programs. We arrange awareness programs. So making society knowledgeable, again, it is our work, right? Our work. And thus, plant diversity is I say, is a biological capital of country. It is a biological capital of the region, and it is a biological capital of man. Every great pleasure of life, whether it is eating, whether it is a nature, scenery, whatever it is, it comes from biodiversity. So it is the only true capital for everyone, and it is the capital needed by every man. So biodiversity basis. Now we have biodiversity information and knowledge created by educational and research institute. That is the basis for other organizations to work. Leaders, lawmakers, policymakers, they use our data. Utilization of biodiversity is our research scholars, researchers, they day and night work, and we use our natural resources. Declaration of mega diversity countries, hotspots of biodiversity, endemic plants, protected area, all that is done on the basis of data collected by educational and research institute. That become basis. Conservation of bioresources, again, that major role comes from educational institutes. Endemics, threatened plant species, methods for conservation, method for propagation, nutrient requirements, and so on, ecology, all that data is created by educational institute. From this figure, I want to tell that educational and research institutes are of great significance. The data collected by them is the basis for every other organization, including government, to work. And therefore, educational institutes have great role. Now, species are invaluable. On one side, you see paper notes, and another side plant. Every plant species will weigh heavier than paper notes. And this is the one story where I will end up. See this, Sri Krishna. This is a story. Satyabhama tried to weigh Krishna with gold. All this gold she put in one pan, but still Krishna was heavier. Then Rukmini came, took out all the gold, and then put just one leaf. It is Tulsi Patra or any leaf of plant. It is an indicator of plant. One leaf of plant was heavier than Krishna. What is the moral of this story? What we want to, what our forefathers have told us. I have analyzed scientifically that our forefathers have told us, oh man, if there is something as important as God or more important than God, it is a plant. And that's why one leaf of plant way heavier than God Krishna. This is a story from our own country, and one should understand its meaning. Okay, plants are the best friends. I don't think anybody of you will deny. They are best doctors, they are best artists, they are best designers, they are best engineers, they are best economists, they are best technicians, and finally they are the best teachers in our life. And therefore, here I want to tell you our evolution. I will not go to all this, but finally about 2 lakhs years before we originated in a Rift Valley, and from that Rift Valley we have migrated to various parts in last 50,000 years. All that, and our agriculture started 10,000 years before, and today we have claimed more than 2/3 land for cultivation, for agriculture. Now remaining 1/3 is left for other organisms. You tell me how much destruction we have done on this planet. If we want to save this planet, we have to do something. Now question is for all of us is how, in what form, whether in a decent good form, we are going to hand over our Mother Earth to coming children, generation, or in a denuded bad form, we want to hand over our Mother Earth to children. Therefore, conservation is the most important part which academics, research scholars, students can do, and there are so many colleges, so many universities. Definitely we can, each university, each college, if conserves one plant species, all angiosperms can be conserved in without money also. So conclude this one, biodiversity is the basis of life. All of you will agree. Role of educational and research institute, teaching, training and research, which becomes basis for everything. Documentation, bioprospecting, utilization and conservation. Major share is done by educational and research institutes. And protecting Mother Earth and welfare of mankind is our responsibility, or we have that knowledge, so we must work. So whatever work our student do, and every teacher should, teaching job is a noble job, and really student is his wealth. So my student, my strength, and through that I could do something in my life. Thank you very much. I am thankful to organizer for giving me opportunity to share what I want to say about our educational institutes and research institute and their role today. And we have to play, or we are the major players in shaping our career.
Thank you, sir. Professor Eshad Yadav.
Yes, sir.
A famous scientist and taxonomist, as we all know. We were very much delighted by seeing the beautiful photographs of the architecture of the flowers like Barleria and Aroids. And he also giving us some information about the monotypic species of Western Ghats and interesting information about Crinum maravillicum and its bioprospecting. He also mentioned the importance of fodder, food, and for other purposes, what is the necessity of taxonomy and documentation. And he also, his documentation of ornamental plants and the domestication of ornamental into his lecture and shown the beautiful pictures of Ceropegia. I can recall in the few months back only we have recorded only one Ceropegia hitsuta, very small herb from the Ajodhya hill. And it was my teacher, Professor M.N. Shannal, who wrote the book Flora of Bankura district, always reminded me that you see the photograph in the first page of the book of Saldana and Nicholson, where you will get the picture of Ceropegia. So I can recall my teacher's words also by listening the beautiful lecture of Professor Jyadav, and also we witnessed the beautiful botanical garden what is constructed inside the Shivaji University. And sir, you rightly pointed and timely, it is your timely lecture, and only the layman can say, only the little known person can say that taxonomy is a dead subject, it is the basis of all subjects. And you rightly pointed out that it is not the forester, it is our duty, the duty of the academicians, the duty of the research institute to make a document not for paper publication, but for the sake of the science. It is the duty of documentation and conservation of biodiversity. Sir, namaskar. It is my great opportunity that I met you through this webinar. So thanks to the organizer. Sir, please welcome to our institute also.
Thank you, sir. Thank you very much.
Thank you, sir. Our next speaker, I am requesting our next speaker, Professor Sheikh Bakhtiaruddin. Professor Bakhtiaruddin was born Kushulia in the district of Satkhira, Bangladesh. He received BSc Honours and MSc degree in Botany from the Department of Botany, University of Chittagong, Chattogram, Bangladesh. In 1993 and 1995, respectively, Dr. Bakhtiaruddin started a Flora 1 Ministry of Environment and Forest, Bangladesh, and began his specialization in the field of plant taxonomy and ethnobotany. In 1997, he received CICHE award for PhD through Aberdeen Chittagong University Biodiversity Link Project. Dr. Bakhtiaruddin awarded his PhD from Department of Plant and Soil Science, University of Aberdeen, Scotland, UK in 2002. He is the first in Bangladesh who awarded PhD in the field of ethnobotany. Then he left for Canada in 2002 as an immigrant and stayed there for 9 months. In June 2003, he returned and joined University of Chittagong as an assistant professor, then as a professor, promoted to as a professor. He has published 4 books, 1 book chapter, and 53 articles. He is the live member of Bangladesh Botanical Society, Biodiversity Research Group of Bangladesh, Bangladesh Association for the Advancement of Science, and member of Bangladesh Association of Plant Taxonomists, Society for Medicinal Plant Research, Germany, International Society for Research on Natural Products. So we are very much curious to listen, and he is delivering a lecture on documentation guidelines of traditional medicine. Professor Professor Bakhtiaruddin, please.
University of Chittagong, Chattogram, Bangladesh. Can you see my presentation? Anyone, please?
Yes, it
Yes,
is
yes,
visible,
doctor.
sir.
We can.
Yes, sir, it is visible.
Can you see?
Yes, yes, it is visible.
Hello?
Yes, sir, it is
Oh,
visible.
okay, is it visible? Thank you, thank you, thank you. So,
Sound is interrupting.
I'm from Chittagong University. It's my beautiful campus of Chittagong University and my lab. So I'm going to talk on documentation of traditional medicine. Uh, we- okay, thank you. So you'll know what is traditional medicine. Traditional medicine describe a group of healthcare practices and products with a long history of use. It frequently refers to
Hmm. Professor Bakhtiaruddin, are you there?
medical knowledge.
Hello. Yes, sir. Sir, it is not audible. Maybe due to network. Uh, it is not audible. We can see the slides. Rest, I don't know whether rest are getting the sound or not.
So not getting, sir,
Then
not
refers
audible, sir,
to
and a little bit of congestion in the
verbal
microphone.
strategy.
Yes, yes, that.
It's used to integrate the different components of the study. So from research design, we have to select all these things, participatory approaches. Who will participate in your research? The obtain permits and permission from the authority. Then we have to get preparations for the field. So there are some official formalities like intellectual property rights, getting permit and permission, and so on. Then we have to take information about the transport system of the study area, then accommodation where we can stay in the study area, and safety and security of the study area. It's very important, and social status of the study area. Then we need to contact the informants, the who will our informants, where do they stay, and we have to collect all the information about the informants before going to the field. Then documentation, information how to collect. We should have regular visit of a study areas. So we have to prepare a visit plan according to the availability of the informants, availability of the flowering seasons. So from the checklist, we can sketch a visit plan in which season we need to visit more and we need to stay more. Then we have to interview the informants for plant use information and record them. Documentation of plant use and collection of voucher specimen. Voucher specimens are also very important. So from the voucher specimen, we have to prepare herbarium. Then we have to identify, labeling the voucher specimens, and then analysis of data. So during the interview, the information has been noted in documentation data sheet. I'll show you a documentation data sheet. And in addition, audio video recording has to be made with a digital voice recorder, or nowadays we can do it with the smartphones. Or have been used for interviewing to gather ethnobotanical plant information. These are the mostly recommended method for ethnobotanical documentation or traditional plant use information documentation. These are direct or participant observation, field interview, plant interview, group interview, checklist interview, and market survey. So,
Sir, your presentation is not seen.
oh,
Your presentation is not seen. Hello? Sir, your presentation
yeah,
is not visible.
okay. Window, share. Is it visible now?
No, sir, not yet.
Not yet.
Not yet, not.
But I'm going through entire screen,
Yes,
share.
right.
Sir, lot of interrupting noise in the background.
Dr. Bhaktiyarudin, what is the problem? Please let us inform or share it. Dr. Bhaktiyarudin, can you hear me? Then I am requesting organizer to communicate over phone or something else.
Sir, I am just trying to communicate Dr. Bhaktiyarudin over phone. I request the participants to kindly bear with us for a few minutes until I talk to him and I let him know what is happening.
Okay, okay, thank you.
Yes, sir, okay.
Hello, Professor Bakhtiaruddin. Are you there? Can you hear me?
Sir, it is not audible, Professor Batiaruddin. It is not audible.
Professor, you need to unmute yourself, please, so that it is audible.
Right.
Can you hear me now?
Yes. Yes. It is audible now.
Okay. And presentation?
Not. Right. Now it is visible. That again, it is lost.
Oh, okay. I cannot be good. Go for.
Yes.
Okay, now. Is it visible?
Yes.
Okay.
Yes, we can see the presentation.
Okay, okay, thank you. Sorry for the interruption because I
Okay, okay, okay. No problem,
lost
sir. Please.
internet connection. So documentation guidelines. I am from Chittagong University, and this is my beautiful campus. So I have already discussed about traditional medicine, and then how to start. We need to do first the literature survey. It's from the book or journals or articles from different journals or internet. There will be different sources for literature survey, and then we have to develop the hypothesis, and from the hypothesis we have to create a research design, and we have a research objectives for documenting medicinal plants. Are we going to collect information for any specific disease or particular group of disease, or in general it includes different types of disease? So from the research design, we have to know the participatory officers who will participate in my research work. Then we have to select the informants who are going to be our informants for this particular research. Like for medicinal plant use information, they might be Boddo, Kobiraj, or elderly people from the study area who have knowledge about medicinal plant use information. Then we need to think about the compensation of informants. There are many ways that we can compensate the informants. So depending on the situation, we have to think about the compensation of informants. Then intellectual property rights is very important. So we have to work on intellectual property rights from the information which are going to study. So intellectual property rights is very important now. And timing, we have to select our field dogs according to flowering times and reachable of the study area, and so on, and obtaining permits and permission from the authority to go to the area. So preparation, we need some preparation before documenting. Just before going to the field, we have to collect some information, because if we work in a systematic way, it will cost less time. So official and legal formalities, we have to know all about this. For that area, transport facilities for the study area, accommodation, and safety and security in Bangladesh, India, and some areas, the tribal area, that are not always safe and secure. So we have to collect information of that area before going, and social status of the area, and then we need to contact the informants. So how to collect information? Regular visit of the state area. So we have to prepare a visit plan according to the flowering, fruiting season and accessibility of the area. Interview of informants about plant use, and we have to record it. Documentation of plant use and collect of voucher specimen. Preparation of herbarium. Identification, labeling of voucher specimens, then analysis of data. So during the interview, the information has been noted in the documentation data sheet, and then audio video recording has been made with a digital voice recorder, video camcorder, or nowadays a smartphone is a very useful tool. And several techniques have been used for interviewing, gathering information. This is direct or participant observation, field interview, plant interview, group interview, checklist interview, and market survey. In direct participant observation, the researcher need to stay with the community and to observe how they are using plants, and we have to collect information from the direct observation. This is direct or participant observation. It is the best way for collecting information, but in some cases, like if there is a safety and security problem, we cannot stay there for a long time, or sometimes for the cultural gap, the community, they don't allow us to stay with them. Then field interview. Field interview, that means the informants will accompany the researcher in the field, and from the field, the researcher can collect information about the plant use. Then plant interview. In sometimes there are some elderly informants, they can go to the field. There are some informants, they are very busy in their profession, so they cannot go to the field with the researcher. So the researcher then collect plants from the field and take it to the informants. They show the plants to the informants, and the informants then give us information about the plant use. Sometimes we can show the photographs of the plants, or we can show the plants picture from computer, laptop, tab, etc. So this is all about plant interview. And then group interview. So in group interview, we can gather many informants together, and from there we can collect information. But sometimes it's very difficult because there are some trade secrecy among them, or social secrecy. So in front of one another, they cannot wish to share their information. So sometimes it get problems. But if it's successful, in a single room, you can collect lots of information from group interview. And checklist interview, from the checklist showing by from the local name of the checklist, we can collect information from the informants. And market survey is a very good for initial survey, because in the market there are many informants, there are many herbal shops, and there are many people, they bring plant materials in the market, in the actually in the tribal market. So it's a very good source for collecting primary information and to locate the informants. So these are this is a documentation data sheet we can use for documentation. In addition, we'll video record and audio record the information. So there is another way, we have to cross check the information, because for compensation for money, sometimes some information informants give us wrong information. So what can we do? We can cross check the information after 2 to 3 months, or 1 month, 2 months, 3 months after the collecting information. We can go back to the informants and ask them about the information again. Then if there is any anomalies, we can find it out. And then we have to we don't need to forget the following information of medicinal plants during documentation. That means the plant parts used in herbal formulation, that means is it whole plant or roots or rhizome or nodule, bulb, comb, stems, and so on. So we have to take all the information, which part they are using, how did they prepare, that means herbal formulation, is it capsule, pill, syrup, juice, leavened, extracts, tinctures, etc. So we have to take all the information. Then how do they preserve? What is the method of preservation? Do they add any ingredients for preserving the herbal medicine? And how do they process? And how long it could be used, the duration? So we have to collect all this information. So how do they use? Are they using externally or internally? If externally, in which process? If internally, in which process? And what are the doses and frequency? How many times we need to take the herbal medicine in a day? And how long? For 3 days, for 7 days, for 1 month? So we have to take all the information by using a documentation and data sheet. And how do they collect? Time of the day. Sometimes it is very important. Sometimes some informant say that you have to collect it before sunrise, or you have to collect it after sunrise. There are some scientific basis for their information, because we do use herbal medicine because there are some active constituents in these plants. So some plants may have a lots of deposition of active constituents in their leaf or in their root before sunrise, or some plants deposit lots of active constituents in our collecting parts, leaves or roots or stems after sunset. So time of the day for collection is very important. So we need to document this information from the informants. And season, stage of maturity, and any ritual activities during or before collection, because in indigenous community, in tribal areas, they have some ritual program before collection. And techniques of collection, because sometimes they collect plants in a very sustainable way, in a very conservative way. So it's very important for conservation, because of over exploitation and in the way that we are collecting, that is not correct. So we are losing our biodiversity during collection of medicinal plants. So it is very important to document their techniques of collection. And how do they conserve their plants? Because they are depending on this traditional medicine, and they are using plants for primary health care in their area. So they always try to conserve their medicinal plants around them. So how do they conserve? We need to document them. Then towards final destination, then we need to do from the field, we'll collect the information, then we'll prepare the voucher specimen, we'll identify the specimens, then The next step is data entry and then data analysis. Then if we have any lab experiments, phytochemical analysis, bioactivity tests, and so on, antimicrobial analysis and so on, we can do it in the lab. And from the result, from the previous data, documentation data and lab experiment, we can analyze the result. Then we have to prepare the manuscript for publication. We published it. We can publish it in different forms, and we have to remember that the documentation we are documenting is a historical document and it's an asset of a country and of the world, because the informants nowadays, the younger generation, they are not willing to take their parents' profession. So with the date of the informants, the information is going lost forever. So we have to document them as much as possible, as soon as possible. What are we doing in our field? We are documenting plant use information from the indigenous communities of Chittagong Hill Tracts, Bangladesh. There are three districts in hilly area of Chittagong, that is Bandarban, Rangamati, and Bandarban, Rangamati, and Khaglachuri. There are 12 indigenous communities living in that area. So they are now, then they are using plants for their healthcare system. So we are collecting, these are the traditional healers living in that area and in the market. So this is our observation, direct observation. They are using the plants. We collect plant and process it. Then the results of our study up to date, we have collected more for 1348 medicinal plants, and we have documented them, and we have published in two websites, Medicinal Plants of Bangladesh and Bangladesh Ethnobotany online database. From our data, we have found that 250 species are currently using for preparation of commercial herbal medicine in Bangladesh. So the result, the study results a total of 1348 species, whereas 1274 angiosperm, 65 pteridophytes, and 9 gymnosperm. Then the pteridophytes, it is angiosperm is 94%, pteridophytes is 5%, and gymnosperm is only 1%. And in our country, the diversity of gymnosperm is very low. There are only 20 gymnosperm present in our country. The study of habit of the medicinal plants that we have recorded, herb is 544, shrub is 272, climber is 201, and tree is 331. Total 1348 species we have been documented using for traditional medicine. The represented family according to number of species found in the family: Fabaceae, Rubiaceae, Euphorbiaceae, followed by Asteraceae, Lamiaceae, are the dominant families according to number of species. And most of the family, the dominant family, contains active constituents. We have surveyed many literature and found that the Fabaceae, Rubiaceae, Euphorbiaceae, Asteraceae, Lamiaceae, Gingividaceae, they are very rich in active constituents. So this indicates the selection of proper families of medicinal use records in Bangladesh. Represent the traditional medicine plants of Bangladesh flora. We have compared in Bangladesh flora from our publication. We have seen that there are 5,016 angiosperm, 21 gymnosperm, and 311 pteridophytes in our country. So the medicinal plants in percentage to our flora is 71.23% angiosperm, gymnosperm 57%, and pteridophytes 87%. It's representing the total flora of Bangladesh. Also, traditional medicinal plants, prominent regions in the world. I have shown it in a table that in China, Sri Lanka, India, Pakistan, North America, Nepal, Mexico, they are using a huge amount of medicinal plants flora. Most of them are native species and some are exotic. So they are using a lot of medicinal plants in different parts of the world. So the using part of the plant, mostly leaves are used for the preparation of herbal medicine, then underground parts like root, rhizome, comb, bulb, etc. Then stem followed by whole plant, fruit, seeds, and others also. So medicinal plants and the Bangladesh economy, 12,500 tons of dried medicinal plants material produced in Bangladesh is sold. $4.5 million to the rural economy and $5.8 million at the factory rate wholesale. And the 5,000 tons of imported medicinal plants cost around $8 million. So we have to import a lot of medicinal plant from India, Nepal, and China. So from our findings, we did some publication. This is about medicinal plants used by Chittagong Hill Textile. This is also ethnomedicinal plants of Bangladesh. We have published it in collaboration with Korea. And this is the dictionary of plant names, where all the plants of Bangladesh have been documented or listed. So we have published 2 databases, medicinal plants of Bangladesh from our lab and Bangladesh ethnobotany online database. So that's all from me. Thank you very much.
Thank you, sir, for your nice presentation. And we have learned a lot about the ethnomedicinal plants and their preservation in Bangladesh. You very nicely explained the different steps and methods of documentation, which informations we have to gather, information about the plant part used, how they are collected, and how they are prepared, how it is used, its dose, and how far it can be used, which preservative is used, and many more things which will be very much helpful to our researchers. And you mentioned some biodiversity hotspot in your region like Bandharban, and it is famous because several videos uploaded in YouTube regarding Bandharban, and we have seen the beautiful vegetation around the streams. So anyway, welcome and very much thankful to you for spending some time with us. If possible, come to West Bengal and visit us, our university. Welcome you, sir.
Thank you very much for your. Hopefully I will be visiting in West Bengal.
Yes, sir.
Thank you. I'm sorry for the interruption because of my internet connection problem.
Okay, that's no problem, sir. That's a technical part. And if any question from any audience, you can ask, because it is an opportunity to learn something about Bangladesh. So if any participant want to ask any question, he or she can ask. Okay. Thank you. Thank you, Dr. Bakhtiaruddin. Any question? Okay.
Thank you very much for giving me the opportunity to present here and to share my knowledge and findings. Thank you very much.
Thank you, sir. Our next speaker is Professor Premlal Unial, and he is the professor of Department of Botany from the University of Delhi. and he has 26 years research experience. He has published one book and author of 3 books. He has published 102 research papers, review papers, and he has published 30 articles, review articles, and research work in edited books. He has published 22 popular articles, which is the need of the day, and he has presented his findings in 50 national, international symposium. He has presented 30 papers in national symposium. He supervised 12 scholars for PhD award, and he also engaged in several research projects. In last 5 years, some major projects are studies of reproductive biology of some economically important farms of Western Himalaya, and preventing extinction, improving conservation status of threatened plants through application of biotechnological tools. Another project was diversity and performance of key forestry species and bryophytes in gap and non-gap areas along an altitudinal gradient in Uttarakhand. And another project, he completed diversity and he is the members of the several learned bodies and societies of national and international repute: International Association of Bryologists, International Society of Morphologists, the Indian Fern Society, Society of Plant Reproductive Biologists, Indian Bryological Society, Delhi University Botanical Society, and many more. He is mainly interested in certain fields of research, and I can mention a few of them. He is studying systematics and ecology of bryophytes, gametophytic development pattern in ferns, reproductive biology of ephedra and taxus, developmental biology of Podostemonaceae, and propagation and conservation of medicinally important threatened plants, investigation on the biologically active compounds from lichen. So we can easily understand from his findings that how much diverse field he covered, which is the indication of a genuine botanist. We are very much pleased to be here to listen his lecture, and fortunate enough, and he will deliver a lecture on strategies of the conservation of medicinal plant. Welcome, Professor Kunial, and you can deliver your lecture, please.
Thank you very much, Professor Raha. I am really grateful to the organizers of this grand symposium for making me a part of this conference, this grand academic event. And there are so much of diversity in the fields, and the speakers are brilliant and from the different subjects, and definitely I'm sure that the audience will be benefited from these diverse fields. So it is great to see the majestic gathering of teachers, scientists, and students, and I really thankful to the gathering for coming here today, and I am sitting in front of you. I am going to share some of the exciting information on the strategies of the conservation and restoration of threatened plants, especially medicinal plants, which is the foremost and priority topic all over the world. I'm sure you will enjoy. So let me share my slides.
Dr. Bhaktiar, if you can stop presenting, if you can hear us.
Yes, yes.
Thank you, Doctor. Dr. Bhaktiar, can you hear us? Perhaps you're on mute.
Dr. Bhaktiar has to
Abhishek, can
leave
you?
the present.
Yeah,
Yeah.
Abhishek,
Yes.
can you stop the presentation from your end? Is it possible to
No,
terminate it?
I don't have that option, but Dr. O'Neill will have the option to like present, start sharing.
Okay.
Uh,
Dr. Dwaipayan, is it
If you
possible to
start
call?
sharing, then Dr.
Yeah.
okay, I am trying.
Thank you, sir.
At the bottom of the screen, there is a share screen.
Okay.
You need to click that, and then your entire screen.
Yes.
Yeah. Now your screen is loading. Okay. We can see your screen, sir.
Okay.
Thank
Can
you.
you see and can you hear me very well?
Yes,
Loud and
and clear.
Okay. Okay. Thank you very much. So, mm, as you know, the conservation of plants, even at the species level, at the genus level, at the ecosystem level, it has become very essential all over the world to restore these species, to reintroduce the species, to gain the native ecosystem for having the ecosystem services and for having the smooth flow of ecosystem services. So, as you know that currently known, described and accepted number of plant species are approximately 3,74,000, of which approximately 2,95,000 are flowering plants, and uh, others are the bryophyte, endophyte, gymnosperms, angiosperms, including all this. So, and it is very interesting that nearly 2,000 species are described every year. So, still, the number of species will keep on increasing whenever the new areas are explored and new species are found. Now, importantly, the diverse group of these plants as well as the animals balance nature and the life on Earth. The species are actually the building blocks of the Earth support system. They are providing several services like capturing and storing energy, providing food, regulating climate,
What is your problem?
decomposing organic matter, cycling water and nutrients, controlling erosion, controlling pests, and this diversity provides a significant environmental and economic benefit to agricultural and natural ecosystems, including adding the diversity and productivity to the food crops, which is very, very important for the human being. So, just I wanted to explain that what is the cost of this biodiversity and
Hello.
what is the cost of.
Kindly make full screen.
Okay. Okay. So, in India, nearly 2 billion tons of food products are harvested annually from the terrestrial wild biota, including large and small animals, fruits, and which is valued $17 million. And part of it add to the India's economy. Medicinal plants and manufactured pharmaceuticals totally rely on biodiversity. The outdoor tourism industry rely on biodiversity that tourists come to see. The diverse microbes fix nitrogen for the use of crops and forest, as well as the vegetation of the diverse biome, and nearly 90 million tons of nitrogen is supplied each year by nitrogen fixing microbes. Do you know this is the free of cost nitrogen the nature is getting worldwide, and the cost of this is $5 billion. Can we produce nitrogen of such magnificent amount? Then nearly $20 million annually is spent worldwide on pesticides, but actually the pesticide this predators existing in the natural ecosystem provides an estimate 5 to 10 times amount of the pest control. Means there is a natural pest control. We are just controlling a small part of the pest, but it is actually the automatically controlled by the nature. And without the existence of natural enemy, crop losses by the pest in agricultural and forestry would be catastrophic, and cost of the chemical pest control would be very heavy. So these are some of the examples. Otherwise, if we see, go deep and count product by product, the cost will be high. So now this is one thing. This is the question on the ecosystem and stability. Actually, a comprehensive understanding of the biodiversity and ecosystem functioning need long-term observations on the change of plant diversity and effect on nutrient cycling and accompanying plant microbe interactions. The concentration of the quantitatively most important nutrient ions such as nitrate, phosphate in the soil solution decrease with the time. Decrease because of the disturbances, because of the ongoing removal of nutrient by biomass harvest. So there is a shift in the microbial community composition in that area, which is triggered by an increased availability of plant derived carbon, suggesting that plant communities compensated by nutrient losses by stimulating the microbial nutrient cycle. So it furthermore employs that plant species richness maintain plant microbial interaction. So what are the consequences? Everyone know this fact that but if we minutely see at micro level, the ecosystem maintain the functioning under various varying circumstances like stresses or disturbances, and ensure the delivery of services because of the biodiversity act as a buffer, as a resilience, as a stable wall. So as ecosystem diversity decreases further and a certain threshold is crossed, the loss of species may result in a sudden and drastic decrease of ecosystem functioning and cause catastrophic ecosystem change. So loss of species across trophic level may alter the food web interactions. This is very, very clear. And decreasing the key mediators of the ecosystem functioning and loss of organism with specific functional traits may cause the direct reductions of the ecological processes relevant to ecosystem services. So this is a very important fact, and if we just see at the macro level, okay, this plant will go, though this will happen. But what will happen at micro level? If you see at different trophic levels, if you see the what will affect, what will happen to soil biota, underground biota, what will happen to detritus food chain? That is another a big question. If there is a species loss or community loss or biodiversity loss from one area. So, in this regard, we need to understand the functional group, species functional group. The importance of biodiversity increases further when we consider the multifunctionality of ecosystem at increasing spatial and temporal variability. Species that control ecological function in a particular year or location often differ from the that control the process of other year or other locations and under other environmental conditions. So the provision of ecosystem services and function is likely to be related to the distribution of these species as functional group. So one should know the category of species. Which species, what is the function of this species? There are large trees, there are shrubs, there are ground layer of bryophytes and pteridophytes, there are microbes on the soil surface, there are fungi and underground microbes. These all make a very, they have a special traits, they have a special function, they have a special attributes. So we should know the category of species or species attribute that can be related to the function. Some groups of dominant and minor species within an ecosystem are functionally similar, and this functional similarity provide buffering or resilience against perturbance or ecosystem variability. So thus the species that dominate under a given set of environmental conditions serve to maintain ecosystem function under those conditions. On the other hand, minor species will functionally similar to dominant species, but in different environmental, but with different environmental requirement and tolerance, and these species maintain the resilience in ecosystem by allowing the maintenance of function under changing environmental conditions. So consequently, to ensure a stable supply of ecosystem function and services, so the conservation and restoration of species through determining the ecological or biodiversity value is very, very essential. So in this context, the concept of diagnostic species composition, a kind of reference state, becomes central. This is most important, and such plant community attributes as the presence of species, abundance of species, dominance of key species, structural or functional and unique element or groups of species that share ecological requirement and features of importance for determining the habitat and structure and composition. So that may be used for practical analysis of plant community quality. So ecological communities or habitat can be powerful tool for achieving cost-effective outputs in land use planning and biodiversity conservation. So we have been observing, I have been working on the conservation, restoration of some of the threatened medicinal plant, especially in the Himalayan region. What I have observed, there is a distinct pattern of habitat modification over the time that have been observed, especially in the Himalayan region. Overall tendency towards a shift in the plant community attributes with lower mean species richness, reduced mean species cover. What I have observed, that increased cover of alien species. I have seen that the Eupatorium and Lantana, they have reached up to 2000 meters altitude in the Himalayan region. How they, because of the how they reach there, because that conditions are not favorable to them, but actually the disturbances in the ecosystem made them to invade these sites. So, so generally, shift in the cover of different groupings. So the community attribute changes and overall shift in the composition and structure of the plant community. Indeed, species loss under increasing land use intensity has already been reported for different species groups. So the disappearance of native species may contribute to making the plant communities more susceptible to the invasion of alien species or weed. So not only the disappearance, but also a small change or decline in the species abundance can lead to the disruption of community structure and also the function. These are some of the factors that increase the diversity. What if there are physically diverse habitat, means there will be more diversity in the species. Physically moderate amount of disturbances, although we need these disturbances, but only the more moderate amount of disturbances. And you know that elephant is called ecosystem engineer. It is one of the ecosystem engineer which break the branches and keep on breaking and which leads to the canopy gap. What happened? Due to this canopy gap, other opportunity species or ground species and seeds which are on the soil may get the opportunity to germinate. So this create the diversity. So this way there are many keystone species that moderate amount of disturbances are required. Sometimes fire, sometimes flooding, sometimes storm, that moderate amount, but not the very heavy amount. Small variation in the environmental conditions like temperature, precipitation, nutrient supply. If there is a, that's why the tropical areas are more diverse, because there is a small variation in the environmental conditions and year-round availability of the water. Then high diversity in one trophic level increases the diversity uh, at another trophic level. Like, for example, many kind of trees provide the habitat for many kinds of birds and many kinds of insects. So ultimately, insect, then finally the pollination. Pollination means very good seed set, and when very good seed set means good regeneration, and good regeneration means a very good vegetation with diversity of species. And middle age succession is a factor for increasing biodiversity. So these are some of the factors that decrease the biodiversity. Everyone know environmental stress. If there is a stress, then very difficult. It directly affect the growth and development of the organisms. Extreme environment, uh, then severe limitation of supply of essential resources. Like if you go to the desert area or cold desert area, there are limitation of the nutrients, also the water limitation of the water, so that cause the that limits the increase of biodiversity. And if there are extreme amount of disturbances, like if you see that if there are frequent fires, then no that once upon a time this will become a desert area, because whatever the seed bank or spore bank that is uh, that is in the found in the underground soil, that will be damaged totally if there is a continuous disturbances like this. If there is a continuous pollution in this water body, there will be a continuous invasion and blooming of the weeds in the water body, and finally this will be a disturbed ecosystem. So these are the some of the factors that decrease the biodiversity. Then here I wanted to tell you that the native ecosystems importance of native ecosystems. Actually, these ecosystems have irrepressible ecological, social, and economic value. In addition to their intrinsic value, such as biodiversity and spiritual or aesthetic importance, healthy native ecosystem assures the flow of ecosystem services. Native ecosystem can reduce the effect of natural disasters and mitigate accelerated climate change. So ecological degradation diminish the biodiversity, functioning, and resilience of the ecosystem, which in turn negatively affect the resilience and sustainability of social ecological system. So very important that why there is always stress, plant a native species, bring back the native species, bring back the native ecosystem. Actually, this is the importance of native species, native ecosystem. So to sustain the flow of ecosystem services, ecological restoration and species recovery program are taken into consideration. So this is just an example taken from the Society of Ecological Research. So monitoring of biodiversity has mainly focused on the species level, with the species level assessment of extinction risk having been used to set priorities for conservation. However, I have seen that researchers are making increasing use of complementary assessment tools that address the higher level of biological organization, like ecological communities, habitat, and ecosystems. So, in these plant communities and vegetation type represent the key approach for biodiversity conservation and restoration above the species level, but increasingly the species recovery program is very successful if we go by species wise. So uh, so this is important that this is the a series of the restoration program, and this is the finally the fully recovering native ecosystem. So in this I was telling you the native ecosystem, or in this case the traditional knowledge, traditional economic ecological knowledge, traditional medicinal knowledge, traditional botanical knowledge, or any kind of traditional knowledge, which is very important. So traditional knowledge based restoration includes the using prescribed fire and seasonal flooding or to modify the vegetation, and conservation of keystone species to improve the habitat. Actually, the indigenous people have used such practices over millennia to increase ecosystem productivity, raw material for food, fiber, medicine, and any other items which they use in their ceremonies. So, traditional knowledge practices increase biodiversity and improve ecological resilience by creating landscape mosaic. Mosaic means you have a different types of the like in one you have a few agricultural land, a part a very good forest, a part grassland, a hilly area, a water body. So this is a landscape mosaic. And if you have a very good landscape mosaic, the biodiversity definitely will increase. So traditional knowledge can help to define native reference ecosystem and catalyze the restoration by allowing applicable application of cultural practices. So in this regard, we also conducted a species recovery program under the Department of Biotechnology mega project. And in this species recovery program, or we set these objectives: survey of the populations of the endangered species and mapping them by using ecological niche modeling, standardization of protocol for macro and micro propagation. So survey means first we should know what is the population size of this plant actually. In the red data book, many of the species are accounted as data deficient. In some of the species, if you see that in your opinion it should be endangered species, but in IUCN list it is not categorized in endangered. In some areas, the species, like I must tell you that Paris polyphylla, that is categorized as endangered species. But if you go to the northeastern Himalayas, plenty of Paris polyphylla is there. So this is somewhat uh, some uh, uh, some of these information is very disputed. So first of all, we should make a survey, and then when we actually know that this species is rare, this species is endangered, this species is threatened, then we go for macro and micro propagation protocol. Then reproductive biology, study of reproductive biology, to see whether are there any constraint in the reproduction and recruitment of this species. Then this assessment of genetic variability, this is another important aspect. If there is a good genetic variability among the population, in the individuals of the population, it means it is a healthy population. But if the population is homozygous, it is very bad, and this population may vanish anytime. And then finally, when you culture these plants by any mean, macro or micro propagate, you have to reintroduce them in the field, in the protected area, to for their survival. So this was the first targets. And finally we took 4 plants: Pictosporum eriocarpum, Lilium polyphyllum, Ephedra gerardiana, Crepidium acuminatum, Schemia inquitelia. So these were the very threatened plant, medicinally important plants, and I need not to explain their medicinal value. But their populations are drastically decreased in the habitat, and only few pockets are left with the populations of these species. So what we make, I must tell you, this is Professor Yadav. Professor Yadav, Professor Indrajit, somewhere Deepa and Sinha is also behind this group. This is somewhere in 2003 when we were on the field trip with the students in the near Shimla in Himachal Pradesh. So this is the kind of survey. This is the kind of survey. Then survey are different types of survey are there: exploratory survey, reconnaissance, extensive survey, and intensive survey. So exploratory survey are done when a little is known about the location and species. So first we should go to herbarium and literature, floras, and personal information, and then we should set the data that what information is available for the species. Then we go for the for actual survey. So search is done as many areas as possible to create a basic plant map indicating the location of the species and distribution and abundance. Reconnaissance is done when abundance and distribution of species is already known, and so survey are conducted periodically to detect new populations. So in this case, new populations also to be detected. Extensive survey are used when data are required that are more detailed, more accurate information is required at finer resolution. Then intensive survey is used when as much information as possible is needed about the species, phytosociology, scientific interpretation of ecological relationship. So this record detailed information, intensive survey record detailed information about the phenology, floristic composition, dynamic features, land use, elevation, relief, slope, everything, etc. So all these surveys are required on the basis of the information already available from the species. So we did the survey. This is the survey, personal survey, Pictosporum eriocarpum, and we wherever the populations were there, we noted the coordinates, altitudes, number of individuals. In this case, male and female are separate plants. So one male, 6 female. So this is very, very important during the reproductive biology study. And then area of occupancy. And so we had the photographs of this, and now you can see that these are really endangered. This has become really threatened. You can see that the new house is constructed, so this plant will go. The wall is constructed in the ashram, so anytime this will go. The road is constructed, roots are exposed. Anytime this Pictosporum tree will go, and this you can see that this is in the Nainital area, and you can see just growing on the slope. And anytime, whenever the landslide in the Nakot, in the landslide will definitely damage. So in this way, and this plant is highly valuable. The people take fodder. People use this as a medicinal plant. Means variety of remedies are cured, taken from this. So this plant, we recorded the actual data, how much the population is there. The another plant was Crepidum acuminatum. This is earlier known as Melexis acuminata. This is the one of the ingredient of Chyawanprash, and this is considered to be very rare because of the extensive harvesting for commercial purpose, and as well as by local people. So we collected the data on population size of this plant. So uh, this is the location. We found many new locations of this plant. So many new locations. Some of these are from the herbarium, but the populations which are in blue, they are the new populations. Actually, locations of the species were actually established by published papers, herbaria, flora, and actual survey in the new areas. So one has to optimize the time of the year for the survey of the species which is currently present and identifiable above the ground. So time of survey is very important when the species is in flowering, when the species is above the ground, and identification features. So the species is flowering and fruiting out of season, and these features are required for identification. So then we should have, during the survey, we should have, actually on the basis of literature, we should have a map, distribution map, also the soil, geology, altitude, etc. All the information we should gather. So this is information is used to prepare species polygon. So species polygon is one of the very important tool for making the distribution map. So this is another species, Lilium folifilum. This is also one of the ingredient of Chyawanprash, and we found this is. I was telling that this species is not in the list of IUCN. Now, recently, this has been included in the IUCN list, but it was not recently, means 5 years ago. But if you see the actual population, there are very few populations are there in the Himalayan region at the high altitude region. So we also collected about the information, coordinates, altitudes, and number of individuals, and this is then area of occupancy. Also, area of occupancy. Area of occupancy means it is an area within the extent of occurrence or which occupied by a taxon, including case of vagrancy. So this measure reflect the fact that a taxon will usually occur, will not occur throughout the area or extent of occurrence. So this is we use the area of occupancy. So these are some of the photographs of the Lilium polyphyllum in the field habitat, and you can see only some of the are large populations, and there are not more than more than 10 individuals in one population. So this is all uh, in the Uttarakhand area, and these are the fruits. These are the actually this is bulb, and this is actually the bulb which is the use. So people take bulb. This is extraordinary, marvelous, rejuvenating bulb. So it is used as a food, it is used as a medicine, it is used in Chavanpras. So this bulb is used. So when people take bulb, so finally what happen? Whole plant will go. So there is no flowering, no fruiting, and this is flower, beautiful flower. This is the these are the fruits, these are the seeds. Actually, the problem is that very slow regeneration rate. At least 8 months, this plants remain under the snow cover. So and that cold treatment is required for seed germination. So this is a very unique plant, and in such plant are required. This is the another plant, Ephedra gerardiana, which is gymnosperms, and in this case also uh, we made the populations survey, and mapped the populations with individuals, number of individuals, and uh, a number of individuals and area of occupancy. So actually, this ephedra occur in extreme habitat. This is the populations, the pictures of some of the populations. This occurs in extremely arid conditions, mostly occur in cold desert region or dry rocks and soil, arid and semi-arid regions, and it is a great source of phytochemicals, mainly because of the plants have capability to grow under extreme climatic conditions. Actually, those plants which are growing in the extreme climatic conditions, definitely they have some novel chemicals. So this plant is used in the variety of diseases, and when people know this, so the commercial companies or the people, they either pay some money to local people to collect this, or either they themselves go in the field and collect large amount of the ephedra plant. So what happened? That hamper the regeneration of this species. So this is population mapping of schemia encotelia, and encotelia in this case also male and females are different. This is an aromatic herb of I can show you. This is, yes, this is a very good beautiful inflorescence. This is berries, this is the populations in the very remote areas, and actually very interesting that the populations are saved in the sacred groups, because this leaf this leaf is actually, and this flower is actually it is considered to be very sacred. So it is protected in a sacred group. Otherwise, outside the sacred group, you will not find any population in the common areas, because people use the leaf are used as a food, or for medicinal purposes, for chewing, for and for uh, insect repellent. In fact, if you just crush the leaf, it will really attract many of the people standing nearby. So this is a miraculous plant, and this has also become endangered. So we collected the information about the populations. Now, next part was the ecological niche modeling. Actually, ecological niche modeling models are the class of methods that use the occurrence data in conjunction with the environmental data to make the correlative models of the environmental conditions that meet a specific ecological requirement and predict the relative suitability of habitat. So ecological niche modeling are most often used in four ways: to estimate the relative uh, suitability of habitat in the geographical areas not known at that areas, means there is no, if there is no earlier information, we can this ecological niche modeling can indicate the presence of species there. Then second, to estimate the change in the suitability of habitat over a time given a specific scenario for environmental change, to estimate the relative suitability of habitat in the geographical areas, so to estimate the species niche. So this species distribution models are increasingly being used in the environmental management, such as managing endangered species, habitat suitability, and environmental change impact. So this is a very, very useful tool. What is used in this case? It's very easy. There is no statistics required in this. In this case, we have to we have an input of some of the variables. These are called bioclim variables. They are derived from the monthly temperature and rainfall value in order to generate more biologically meaningful variables, and these are often used in species distribution modeling and related ecological modeling techniques. So bioclimatic variables represent annual trends: mean annual temperature, annual precipitation, seasonally annual range, temperature, precipitation, or sometime limit of the temperature or precipitation, etc. So all these we have to we can get this by climatic variables very easily, and if you put them in the software, Jiva DIS Maxent model, you will get the map, and you will get the such graphs, such map area of curve, response curve, and in response curve, uh, this is area of curve that represent the degree of measure of suitability. It actually tells how much model is capable of distinguishing between the classes. Means higher AUC, higher area of curve, means better the model is predicting zero. And so generally the value is between 0 to 1. So I will tell you in the next model. So this is what we, on the basis of our records population survey and the bioclim data, we put that, and in this case distribution of Pittosporum was predicted. And this is a predicted distribution of Pittosporum eriocarpum. In this case, the yellow color. If there is a yellow color, this yellow color indicates the area predicted to be stable for species absence. So no, this species will not grow in the area of yellowness. So orange color, that indicate the area gained by the species. The green color indicates the area to be lost by the species. Yes, this was probably this was the species was here, but this was lost because there was a frequent disturbances in the climatic factors. And this red color indicates the area to be stable for species presence. So you can see, you can predict the actual distribution of species in this area. So Pittosporum, this is the most suitable habitat. This area, probably this is Uttarakhand. and this plant is endemic to Uttarakhand and Himachal Pradesh, only in the lower hills of Himachal Pradesh and Uttarakhand. So this model is very good. Then we put the this ecological niche modeling in the Maxed model AUC value. This has become 6. So it is near 1. Near 1 means this area of curve is very, very good, and this show a very good prediction of this species. So they show the distribution of Lilium polyphyllum in the higher altitudes of Himalayas. So green shows that probably this species was earlier there, but now not possible, and yellow show that definitely absence of this species. So same way we prepare the distribution map of Amphitrachyradiana, and this show the only the higher altitude region, especially the Ladakh region, cold desert, Ladakh, and dry areas of Himachal Pradesh like Lahore and Spiti Valley.
Professor Unial, I'm sorry for the interruption, Professor Unial. Due to some technical problem, we are running late.
Okay.
Okay, please, sir. Please
I
continue.
will. You want me to finish quickly? Huh? Sir, you want me to finish quickly?
If it is possible, sir.
Yes, definitely. So this is again another model for Cryptodium acuminatum. You can see the Cryptodium acuminatum distribution model. Then Schimia inquitelia. This is another distribution model. So from this conclusion, we made this conclusion that because of the developmental activities, because of the population size due to the over collection, and we recorded all the data. And then finally, the second target is that the propagation, micropropagation. So we did the micropropagation or for Pittosporum. So shoot induction, was in MS medium supplemented with BAP, and finally we got the shoot rooting medium and rooting medium by IBA, and finally we got a very good plantation plantlets in culture, and finally the hardling was done. And finally this is the Lilium polyphyllum, and in Lilium polyphyllum we took the scale leaves of the bulb, and finally we got the callus, and these are the plants, and finally we could harden them. So these are the hardened plants in the greenhouse culture, finally transferred to vermicelli. Then this is the Cryptodium acuminatum, and we got a good number of plants by the combinations of the various hormones after the great efforts. And finally we got, this is the Ephedra gerardiana, and we got plants. We made the study on some of the endangered pteridophytes and bryophyte species, and finally we got the very good plant. So in this case, we got the successful micropropagation in Pittosporum, very slow growth in Cryptodium, very slow growth in Lilium, because this is a highly very unique plant, and we don't know what conditions are required. Still, we don't know. Then Ephedra gerardiana, very slow growth in the then reproductive biology. So reproductive biology is highly essential. That are there any constraint in the pollinator constraint? Are there enough pollen there? Fertilization process occurs, and we see that this is the bumblebee in the case of Lilium polyphyllum, and bumblebee population is decreased due to the climate change and shift. Due to the climate change, there is a shift in the phenology and this bumblebee populations. So that is greatly affecting the population. So in the case of Schimia also, we thought what is the actually seed formation is very less in this, because fertilization fail. You can see there are dimorphic pollen. Some of the pollens are, half of the pollen in anther are non-viable. You can see the acetocarbon test, and probably due to the some of the structural changes in the surface of the flower also may hinder the pollination. So this is Ephedra gerarda. In this case, we never noticed any abnormality, although pollination success is there. But in this case, the germination is very slow. So in the case of Crypedium, we also recorded the phenology, and that is very important. Then pollinators, structures, and are there any anything? These are the pollinators, and very surprisingly that now due to the loss of many tree species, the many of the spiders which were occurring on the tree species now shifted to the herbaceous species. And due to the herbaceous species, these spiders are generally, this you can see the spider's net on the surface of the flower, and actually they eat the pollinators and hinder the pollination mechanism. Probably this is one of the reasons that the fruit set is not been successful. This is the in the case of viola, various pollinators. And in the case of viola, we saw that in situ pollen germination occurs because of the stress of the pollinator scarcity at the higher altitude. So there is a in situ germination in the pollen in the chasmogamous flowers. So we also calculated the nectar estimation of the various contents of the nectar, and sugar is very important in this case. If the temperature goes high, the sugars will decrease. So in this way, we concluded that in Pittosporum there is no reproductive state, but only the delay in seed germination. And by the time, because the delay in seed germination, by the time the seeds are damaged by the animals. In the Crypedium, there is a heavy rain because of their canopy gap, because of the early fall in the heavy rain, and constraint in the pollinator availability. So in this case also we concluded some of this. Then we also assess the genetic diversity, and in genetic diversity what we saw that homozygous populations in the case of Rhipidium acuminatum. So one thing is very that probably the plant is propagating by the rhizome only, but thus not by the seeds. Probably this is the reason. So finally, we got it. In the case of viola, we identified very good, a unique kind of cyclotides, and we tested its possibility against the malaria. And finally, we got that this is a very good anti-malarial. It can be very good anti-malarial drugs in this. Then finally, what happened that we cultured, we mapped the populations, and finally, what happened? These are the cultured plants, and finally hardened plants, and they are reintroduced. Pittosporum plants reintroduced in the protected areas in the Himalayan region. They are growing well. This is the another nursery. These are the Crypedium acuminatum. These are the micropropagated plants, and we and surprisingly, in this, what we did that the populations from different locations cultured and they are planted together, so at least we should have the heterozygous populations if the pollination occur in between. So they are growing well now. You can see that a very good populations in the 3 years. This is the ischemia, and this is now the pharmaceutical company need not to take the resources from the natural habitat. This can be grown in the one glass house or greenhouse, and we have the culture techniques, and finally they are hardened. And in the case of ephedra also, these are the hardened ephedra plants, and finally they are in the nursery somewhere in the nursery. There's a hardening process and they are growing well in the nursery, although we are waiting for the own production. This is the nursery in different locations in Uttarakhand. We also did the Texas, and this is the Texas. Texas, we just by the cutting of the Texas, we planted, and this is the Texas plant, and finally new plant comes. Then these are planted in the Pangi Valley, Himachal Pradesh, in with the forest department, with the collaboration with the forest department, and a very good livelihood program can be initiated with the growing of taxa. So we have at least restored some of the populations of the reintroduced, and some have created new populations of this plant. These plants, these threatened plants. So the conclusion is that social and scientific roles of biodiversity are becoming more consistently defined by creating curiosity, highlighting the interesting knowledge about the plants, complement the management and conservation strategies at local and regional scale. And this statement perfectly match with the what Professor Yadav today said. Thank you very much for your patience.
Thank you, Professor Unial. You delivered a very nice and elaborative lecture starting from ecology to micropropagation to reproductive biology, and we are waiting to learn the details from you. If you kindly attend some other webinar conducted by us, then we will be happy to learn a lot.
Definitely.
Very,
We
very,
can arrange
very.
a workshop. Yes.
Yes, so thank you. And particularly, you have discussed the consequences of the loss of species. We understood the species functional groups, the determinant and minor species concept of diagnostic plant composition, habitat modification, and its relation in species composition. We have also learned that sometimes small factors, small disturbances in the ecosystem increase the biodiversity, or increase, like you called about the elephant invasion, how that can enrich the biodiversity. These are very interesting, really interesting information. And you have highlighted the the concept or the importance of native ecosystem, eco-restoration, traditional knowledge, and conservation strategies. We have also learned about your thorough survey work in the high altitudes and ecological niche modeling, reproductive biology, and micropropagation. A lot of things. Arena you covered, sir, in this short period of time. Thank you very much, sir, for attending this seminar and sharing your concept. Namaste, sir. Thank you, sir. Now I am requesting our last but not least, the speaker, Dr. Induchudan, N.C. Induchudan. He is MSc in Forestry from Kerala Agricultural University, PhD in Ecology from Pondicherry University for the Ecological Studies on Sacred Groups of Kerala. Post PG courses he has done in Landscape Ecology from Melbourne University. Professor Indu Chodan, also a former scientist in ecology in Kerala Forest Research Institute, Trichur, and later Deputy Conservator of Forest, Kerala Forest Department. Then he joined as a director of Atapadi Hill Area Development Society. Now he is retired but active as an environmental worker and teacher's trainer. State Vice President of Bharatiya Vidyaniketan and State President of Vidyabhasya Vikas Kendra, working among the teachers for value-based education, what is the need of the day, and he is in charge of environment work in Swadeshi Science Movement, Kerala. Now I am requesting Professor Indu Chodan, Dr. Indu Chodan, to deliver a lecture. The title of his lecture is Man, his connectedness with nature. Sir, please.
Namaste. I'm audible.
Namaste, sir. It is audible.
सकल के शुभकामना हमारा बंगला भाषा कम ज्ञानी बंजारा पेरे निदे पेरे आनंदिता
Thank you, sir.
सुतराम हमारा पेपर इंग्लिश है उपस्थापना.
Thank you, sir. Yes, sir.
We'll start. Yeah, Bengali was my second language.
Oh, that's fine, sir.
Bengali is a second language, so I can read and write.
Okay, sir.
So today our topic is, as announced, first of all, thank you all for giving me an opportunity, especially Doiba and Sinha for contacting me and taking pain for the perseverance. All the learned scholars who have devoted their valuable time for this session, the international seminar to the world of plants. Actually, it is a biological ingenuity, the plants, especially the tropical forest where we are living. My topic is connectedness with nature. We know we are part of this nature, and this nature is part of us. In traditional, our forefathers used to say, Aham Brahmasmi. So they tried to understand the universe. They always try to, they baffled. And they were fascinated by the mysterious ways of nature. And he was baffled at his marvelous arrangements, obeying certain laws. There are certain laws, physical laws and spiritual laws. And all the human inquiries about the origin and nature of the cosmos and our relationship with it have given rise to the religion and science. In the midst of this pandemic situation, COVID situation, it is necessary to examine the synergic effects of science and religion to shield the human body and soul. Only difference is when the Western world started recognizing the presence of a cosmic force in their efforts to understand the nature, the Oriental, the Eastern, especially Indians, they experience this universe as a manifestation of the cosmic force, which they consider divine. Even though they were trying to understand God, actually they were trying to experience Lord. The whole knowledge called Vedas had been generated in the pursuit of understanding this divine. To understand the God better, they looked upon his creations. Entire knowledge they gain is presented along with a mystic experience. And they had to depict a clear understanding though of this universe. The informations generated through modern science has always helped the modern world to realize and appreciate the capacity of the early Indians to understand the world better. I'm not trying to tell the parallelism in the Vedic thoughts and modern science, not to show that everything of the modern science was known to Indians as an intuition, but just to indicate that there are other methods of thinking to arrive at the truths or to generate information about the universe. So there are different methods. So our traditional society both relied on modern science and at the same time spiritual science, Vidya and Avidya. So we realized, we understood that this, we are all on the earth. Earth is a planet. It is around. It's in the solar system, and the sun is just one star in the Milky Way or the Milky Galaxy. This Milky Galaxy has 10 raised to 12 number of stars. If you start counting 1 crore, 2 crore, 3 crore, 4 crores like that, 1 lakh crore, 10 raised to 12. So there are 1 trillion stars in this universe. So our sun is one in that 1 trillion. But this is a small galaxy. There are larger galaxies also. Am I audible?
Yes, sir. Yes, sir.
Doibanji was trying to call me. I will give another number. If some of the organizers want to contact me, 94966 57925. 94966 57925. In the midst of the talk, if the organizers want to contact me for some communications, your Doibayan Sinhaji was trying to contact.
Thank you, sir.
Sorry. Amo. So this galaxy, one among the 1 trillion, but it's a very, very small galaxy. Its diameter is just 10,000 light years. There are larger galaxies also. This is a small galaxy. There are such different galaxies. Like that, there are more than 100 billion galaxies in this universe. More than 100 billion galaxies. So all these different galaxies constitute this universe. Like that, there are different universes also, multiple universe. The modern scientists have also arrived at the multiple universe. Our spiritual books also tell about multiple universes. The specialty of all our study about bios or biology is that, or the environment work is to protect or safeguard life on Earth. When you consider the entire universe, crores of trillions of galaxies, stars, many planets, but we have the modern science know in this universe life is known to exist only in this Earth. That's the most important. So why there is life on Earth? So our ancestors said it is due to Panchabhutas. We are all made of living and non-living things. In this universe is made of Panchabhutas. Agasha, Bhumi, Jal, Vayu, Agni. Agash means space. Space. All matter requires space for existence. Then Bhumi, Earth. We are all the converted forms of Earth. When the trees are standing, it is a converted form of the Earth. And there is water. More than 70% of our body contains water. More than 80% of our blood vessels contain water. Around 75% of your brain, water. Even your bones, around 30% of the bone weight is due to water. So water is an integral part. So if Earth is our food, annam, carbohydrates. Without food, we can live for 50 days. But without water, we can live maximum for 5 days. Next is air. It is more important than water and air. Without air, we cannot live for more than 5 minutes. Next is energy, the cosmic energy, solar energy. Energy or Agni on this earth is from the sun. the role of the trees, all the green chlorophyll pigment on earth is to convert this solar energy to utilizable biomass. So when we stand below a tree, we call it a shade. There is less sunlight, less heat. This heat is absorbed by the plants. So carbon dioxide plus water will give only carbonic acid, but the carbohydrate is generated when 23,400 calories of heat is also absorbed by 1 gram. To produce 1 gram of biomass, it requires 23,400 calories of heat. So that much heat is contained in the plants as carbohydrates. So when it goes to our body, when it is digested, carbon dioxide comes out, heat is also coming out. So breathe out carbon dioxide, heat goes to our body. The heat of all the living beings, all the animals on earth, all the birds, all the worms, this is solar heat. So energy or Agni from the trees. Suppose we are sitting below a tree. How we are connected with the tree? There is no other organism having more connectedness with us than the trees or than the plants. We breathe out carbon dioxide. When we breathe out carbon dioxide, the trees breathe in the carbon dioxide. We breathe out. And they breathe out oxygen. They consume carbon dioxide, breathe out oxygen. That oxygen we breathe in, give out carbon dioxide. Carbon dioxide we give out is consumed by the plants. Like that, if we stand below the tree, or there are some dwellings below the trees, or we are inhabiting a certain area, we are exchanging our materials. We have got a close relationship with the plants. The carbon dioxide we breathe out, it is absorbed by suppose we take some food. Basmati rice is brought from Uttarakhand. The carbon dioxide that has been in the air of Uttarakhand converted to carbohydrate, coming to our plate, we eat it. When we breathe out, the carbon dioxide goes out, is absorbed by the plants, becomes some flower, fruits, etc. Suppose the honey is consumed from butterflies. A butterfly is eaten by a bird. That bird is eaten by a snake. Suppose a snake is crawling on the ground. If we look up observe it with a molecular microscope, we can see the same carbon dioxide. So the same carbon dioxide which was blowing in the air in Uttarakhand, which was in our plate, which we had consumed, which we have breathed out, which was in the tree, which was in as honey, as flower, as fruit, which was in the bird. Now we see the same carbon molecules in the snake. If snake is decayed, become some worms, some plants grow there. If that plant is eaten by some deers, a deer is eaten by some lion, then the same carbon dioxide we can see. So suppose we are inhabited, we are living in a certain area, entire body particles are exchanged with the living beings around. So there is an intrinsic relationship. We are just exchanging the body particles of all the beings around us, the plants, butterflies, birds, animals, etc. So really forefathers used to say, this is your brother, this is your sister, all the standing trees are our forefathers. At first, apparently we may think it is all nonsense superstition, but when we analyze, when we contemplate and observe things at a molecular level or atomic level, we see that we understand there is a truth. So there is an interconnectedness with us. Everything is interrelated. So what our forefathers said, that we are part of this universe and this universe is part of us, is same, exactly scientific truths. So all the organisms are connected. And also, we understood there is a divine guidance for all these animals. All these animals, birds, they can survive in spite of the drastic changes we have made in this catastrophic change on the surface of the earth. They are surviving because there is a divine guidance for these animals. I will just tell an example, which I have seen in Animal Planet around 10 years back. A type of ants in African forest. It was going around 500 or 600 meters away. It collected some egg from beneath the certain leaves. It brought and kept in its hive. Then after some time, the egg is being hatched. Some worms comes. They are milking worms. There are certain types of worms called milking worms. So these ants know this is milking worms. It will hatch. When it comes out, they will consume milk. At the same time, certain ants will go out, bring food for this. They know the particular type of plants which is lactating, which gives more milk. So that is given. And after some time, it goes to pupa. Then it comes out the butterflies. There's a symbiotic relationship. The ants were benefited, the worms were benefited, and the butterflies comes out. So butterfly was benefited because it was not consumed by some predators, because it was safe in the ant's house. So when it goes, so every year, every day, there is hatching of eggs. So these ants are so knowledgeable. They know the touch with their antenna, the eggs. They understand this will hatch on 10th, means it will hatch on 10th. Next batch, it will hatch on 15th, means it will hatch on 15th. Because maximum the life of this milking worms is 7 to 10 days. So just like we are managing a dairy, these ants are capable of managing this milking worms. So where do we get that information? How does this migratory birds comes? Thousands of kilometers they travel. We have made the changes. Now we have got GPS and all. But even without that, these animals, these birds, innocent beings, they fly thousands of kilometers. If you want to send a plane, we have to refill our tank. You have to land somewhere. Transit stations are there. Without any transit stations, they come migrate thousands of kilometers. It is said that there are certain chambers called golden chambers in the mountains itself. There's a golden ratio, 1:1.6. Where in the Himalayan regions and all, if you cross that, if you are inside that golden chamber, there is energy. Energy is created. So with less energy, less fuel, they travel thousands of kilometers. Correct destination, they come, they go back. Intercontinental migratory, there's a guidance. We apply, then this, suppose there is an ant is going, we are spraying some insecticides or pesticides. They realize without consuming there, without going there, without falling on their body, they understand it is dangerous. They don't go, they just avoid. They travel. They travel 500, 600 meters through all these hindrances we have created. Because they know there is a guidance. There is a guidance from within. The dwelling, that normally we say, Andaryam in God. Throughout India, we call Andaryam in God. God is dwelling in the hearts. Like that, these innocent beings are being guided by the divine force. Whatever we may call it as God or the cosmic force, but there is a force guiding all these animals, all these organisms, and we have an intricate relationship with it. These deep links of man and nature were fully understood by the ancient men of India, who considered this entire nature divine. The things that they could not do, they called divine. Everything was mystery for us. From the same soil, a beautiful flower is there. Strychnos, plants like Strychnos nexomica comes. Fruits like apple is made. Fruits like mango, Alfonso mango is generated. From the same plot, with the same soil, same water, under the normal atmospheric temperature and pressure, This is with the difference in cosmic energy with absorbed due to the separate venation and the angle with which it is facing the universe. Like that, the things which if we want to make 1 gram of sandal, we require a plant worth crores of rupees. But with the normal temperature and pressure, on standing on the same soil in the open sky, it creates sandal, it creates jasmine, it creates red sandals. So all these plants are actually wonders. So that which we cannot do, we call miracles. Certain divinity is attributed, but we understand there is some cosmic force behind them. So all these animals, just like human beings, they also want to live happily, freely. But we go and kill them sometimes. None of the animals, none of the beings want to be killed. Nobody likes to suffer physical pain. So when we are going to cut an animal, cut a goat or buffalo, it also surrender before God. So who the blessings of the God will be with us or with the buffalo which is crying, innocent animal crying. And the more important thing is, when such animals about to be butchered, about to be killed, their helplessness, their passion, intense passion released to the atmosphere goes to the photosphere. Just like atmosphere, there is a photosphere also. And these thoughts travel. It enters the new babies also. Enters and comes out as helplessness, revenge, violence, etc. So whatever violence we do to this universe, to do to the plants, do to these animals, it reaches back. It's called karma also. So there is an intrinsic relationship between plants and animals. Everything is interconnected. You sow what you reap, but many times over, it is just like we are planting a mango tree. It grows out. We have planted only one seed. Thousands of fruits we are getting. That fruit, somebody will plant, lakhs, crores will get. So that is a karma theory as generated. It's the best example for the karma theory. Everything what is done to this universe, it comes back. If you throw a stone, it will come back as mountain. If you throw a flower, it will come back as a garland. So we have to be compassionate to this animal. And India is the only country in the world which has written in the constitution that we have to be compassionate to all living organisms. It is because of our culture. So with this compassion, my time is almost only 1 minute more. With this compassion towards all living beings, we should be able to live in this planet, sharing all our resources. It is not only our own, other animals, other species, plants, they are also eligible for this. They have also the right to live, right to survive, which we are not allowing, or which the human species have become a threat to the universe, neglecting that universal wish, neglecting the desire of the all other living organisms. It will have repercussions also. You see the later diseases, more than 80% of the diseases are zoonotic diseases. The health or wellness of other organisms, either rats or bats, even other virus, bacteria, everything affects us. So it's a repercussion or reaction or repellent action result. All the diseases are repellent result of the lack of wellness for other beings also. That's why our forefathers said, Sarve Bisugina Sandhu, Sarve Sundundiramaya. The wellness, the modern theory of the ecologists is wellness for all. Entire wellness, which our forefathers wanted, aimed at thousands of years back. With this, I conclude. I hope I was within the scope of the title. If there is any questions, please. Namaste.
Thank you, sir. Thank you for your insightful lecture. And you have opened up a new dimension, which is completely different from the previous lectures. And we can understand now why you are teaching moral science to the Kerala. And you have rightly mentioned, that is, I agree with you, and Professor Mukherjee is also present here. He is also a philosopher, and we agree with you that Aham Brahmasmi, or each soul is potentially divine. You started with that, a fine and tuned the lecture very nicely, where you have also mentioned that starting from the universe to the food chain to the each and every material is made up of same thing, a new dimension, of course. And you have also mentioned, that is very appreciable, that may not be every time, but intuition is a factor, as evident from the thought experiments of Einstein, Nikola Tesla, and Swami Vivekananda. All their hypotheses came out through their intuition. So you rightly point out, and it is the very, very demand of the day and need of the day, that the modern generation should learn something about this philosophical concept behind biology. So thank you, sir. You also mentioned about garden chamber, the high energy status of the high altitude, and when it decreases, it lowers, and several new dimensions. I think this thought-provoking lecture of you benefits our students. And welcome to West Bengal, sir. Once again, as you have a close relationship with West Bengal, please come once again to our state. Thank you, sir. Thank you for your nice deliberation. And with this first session probably ends, and it is over to the organizing secretary, Dr. Doikan Shina.
Hello, sir.
Yes.
Hello, Professor Raha. Could you hear me?
Yes. It's audible.
Thank you so much, Dr. Induchudan, for your wonderful lecture, which has enlightened us a lot. And I personally now thank Professor Subrata Raha for being kind enough to chair the session since the morning. He has been a great support to us in the seminar, and on a personal level, he has inspired me a lot in conduction of the seminar and has been a good guide of me since last 2-3 days. I personally thank him once again. He's like my elder brother, and he has really supported me in the process. Thank you so much, Professor Raha, for your kind cooperation and your kind cheering session. And I once again thank all the resource person, all the chairpersons, and the special guests who have been there in the morning, as well as the participants of the event. Now we have another lecture in the first technical session. That lecture would be delivered by Professor Ambarish Mukherjee, and he was scheduled to. It was scheduled on somewhere at 1:10. I request the audience to kindly bear with us. Let me contact Professor Ambarish Mukherjee, and I hope we would be very soon connecting him to the session. I request to bear with us for a few minutes. Thank you. Hello. I think Professor Ambarish Mukherjee is with us. Good afternoon, sir. Hello, could you hear me? Hello.
Sir, please
hello,
unmute. Sir, please unmute.
Yeah, sir, you're on mute right now. Just unmute yourself.
Shall I unmute?
Yes.
Hello.
I think he just did.
Hello.
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 I am with all of you.
Professor Tariq Ali, I request you to kindly introduce Professor Ambarish Mukherjee, though he doesn't really need any introduction. He's a renowned figure. If you can kindly initiate the introduction, that would be of great help. And I'm unmuting myself.
Tariq Babu.
Yes, Rajan sir. So, in this session, we have with us as resource person Professor Ambarish Mukherjee, and this session is going to be chaired by Dr. Rajendra Prasad De. I request Dr. Rajendra Prasad De to take the charge of the session and introduce our honorable speaker.
Hello.
Sir. Yes. Good. Yes.
Good
afternoon.
So, best wishes to all of you.
Everybody. Now we reach at the end of the first technical session. The last speaker of this session is the renowned plant taxonomist, Professor Dr. Ambarish Mukherjee. I have nothing to introduce him. He is well known to most of all our audience. Ambarish Mukherjee, a postgraduate Calcutta University, received his PhD degree from Calcutta University in 1981. He is Premchand Raichand Scholar and Moat Medalist of Calcutta University. Professor Mukherjee was declared Fellow of Ethnobotanical Society in 1992, Fellow of the Indian Association of Angiosperm Taxonomy in 2011, Fellow of National Environmentalists Association India in 2011, Fellow of the Linnean Society London in 2012, and Fellow of Global Academic Society in 2015. The Karl von Ossietzky University Oldenburg, Germany awarded a grant for his contribution in the ISEE conference in 2010. He was awarded Gopinath Panigrahi Memorial Award by the Botanical Society of Bengal in 2017 for his contribution in the field of plant taxonomy. Professor Mukherjee is in teaching profession since 1979 with great reputation. When he joined the postgraduate department of botany, Darjeeling Government College, he had served the UGC Center of Advanced Study, Department of Botany of Bardhaman University from 1993 to 2017. Professor Mukherjee has wide and varied interests and active involvement in plant taxonomy, ecology, environmental science, ethnobotany, especially tribal medicines and pharmacognosy. Under his able guidance, 24 research scholars have received their PhD degrees. 10 students completed their MPhil dissertations under his guidance. Professor Mukherjee has published 6 books and over 250 papers and review articles in reputed journals and books. His academic visit to China, Germany, France, Austria, Czech Republic, Bangladesh, and United Kingdom were worthwhile in fetching him academic excellence and reputation. His association with the Natural History Museum, Erfurt, Germany, for the last 8 years is noteworthy. The title of his lecture is With Plants Down the Memory Lane. Now I request him to deliver his valuable lecture. Sir, please, sir.
Can we start prayer? Please bear with me. I am unable to connect to the PowerPoint. Hello.
Hello.
Are you able to see the PowerPoint presentation?
No, sir. Actually, you need to share the screen. So there will be an option at the bottom right hand side. Present now.
Please bear with me. Huh?
Yeah, sure.
Share.
Yeah. So please click on present now, and then you need to select the option of entire screen.
Share.
Please click on OK.
Should I click on share?
Yeah, please.
And I don't feel.
So in case the share option is grayed out, you need to select from the drop down as entire screen, and then the share option will be enabled.
Are you able to see?
Not yet. Have you clicked on present now?
Yes.
Okay. Then what option do you see? There will be two options: your entire screen and a window.
Just share is one of the options.
Yeah. Now we can see your screen.
You can see the screen.
I'm able to see your screen.
So this is my topic which I'm going to present.
Sir, request you to put it to presentation mode so that it will be visible for all. Yeah, thank you, sir.
So the topic is already declared, announced, and let me first thank the organizers for just giving me an opportunity to be with my students and also friends, and also the academicians coming from different scientists coming from different parts of our country and outside. So the wonderful theme of the webinar, this world of plants, its impact on indigenous people, society and biosphere, has most charming insights me to watch.
Hello. Kindly hide notification panel. Kindly hide notification panel.
I don't understand what it says.
Hide, sir. There is a strip. Hide, sir. Hide, sir. Hide. Thank you, sir.
Okay. Is now okay?
Yes, sir. Now it's okay. Please
So,
continue.
you know, since I retired last year only from Kalyani University, I was a visiting professor. And then after that, I had been to Chennai, and I mostly travel to different parts of our country every time. Every year, I move from here to there. But you know, I had been to Chennai this January. Then I went to Vrindavan in March. And then after that, I got confined into my own study room, and I got no scope to go outside even. So that put me into the memory lane. And I was just recapitulating what I did and what I obtained from the plants. So what was my realization, you know, that whatever I have studied from the university, It was, in fact, inadequate. Whatever I learned, there was from the people of our country while traveling to different parts of the world, different parts of my country, during my professional career as well as during my research tenure. And I came very close to the people of India, mainly the tribal communities, from whom I've learned a lot. So, firstly, I must say, while traveling along my memory lane, with plants, of course, I will first of all pay my tributes to such resource persons, the traditional knowledge holders of our country. Thanks to them for giving me so much of say charm in my professional career and also so much of peace and bliss in my professional career. And I must thank my dear speaker for such an excellent feelings about the say the divine botany. I must say that was a separate subject. How nicely he could place before us his feelings, and also thanks to Hindu children for such an excellent humanity that he has shown within him. So thank you, Dr. Children. And now, what I would like to say is, while traveling with the plants down the memory lane, I just reached the Vedic era. Of course, and what I picked up today is to speak on the ancient say, period, whatever knowledge we had in the Vedic era, especially, and then also the contemporary revelations. How much do we still retain the knowledge that we acquired, that we had, that we possessed during the ancient times? Of course, I picked up the Vedic period, from just show how rich was our knowledge about plants, and in every discipline of our botany or plant science, we really had mastered and gathered a lot of knowledge and resource. And that must be, today whatever we have achievements in our field of plant science is deeply rooted in our achievements and our scientific endeavors in the ancient era, especially in the Vedic era. So that I shall concentrate upon the plant knowledge in plant science during the Vedic era, and also what survives today among us. So, firstly, let me pay my tribute to my nation, because I've learned a lot from the people of our country. Well, as I've already admitted, that I've learned mostly from the people of India, residing in the cradle of nature, sharing the moments of their joy and sorrow with the nature. And it's there, because of them only I could come very close to my countrymen. My country is such a great nation, and so I always will be feeling in my duty if I don't start by paying my tribute to my nation, such a great nation. Here for a while is certain to glorify life. So it's a country of great cultural heritage, the land of eternal peace and joy. But the main motto of social life is to live in harmony with nature. The traditions still enshrine respect for nature and consider its protection as sacred duty. And we have the great geographical and ecological diversities in India, nurture a rich diversity of living organisms. And even you will find here songs are tuned up in the rhythm and musical notes of nature, planets, mountains, hills, air, water, soil, fire, earth, worship. So we have the great Himalayas in the north, a blissful divine abode. So with the plants I was traveling far and wide, and the places which I traveled, and I came across different plant communities as well as human communities. Such a wonderful association of man with plants. So down, and also how the biodiversity, especially the plant diversity, had its impact on the human society, their feelings, their values, their ethics. So wonderful, say, kind of understanding and wonderful kind of coexistence that still persists in India. So India is a great country that can give leadership to the remaining parts of the world to even shift from what it is, this superficial ecology to deep ecology. I can speak on deep ecology, where it is advised that one must spend some days in India to have a change, transformation from man to human. So it's the religion of humanity that still persists in India, that exists in India, that can bring about a change in our outlook, in our ethics, in our values, and give the world a different shape and take us to the days of peace and bliss. So just downstream tributaries of Indus along the Ladakh border, Himachal Pradesh, Ladakh border, glorified India with civilization of the world. Seen silently, the greatest pilgrimage of no clients still bear it. The feeling of Maurice God, the God still exists. The Sapta Sindhu, on which the world's most ancient civilization, the God is here.